Hello, my dear students, and welcome back to Victory Batch. And I am your Diksha ma'am. So, today we are going to start with another very important chapter from human physiology, and that is neural control and coordination. I hope a lot of you, find this chapter to be very difficult or very lengthy. Things are very different. There is nothing like that. Today, I will prove you that it's one of a very interesting chapter because this is the only system that controls your entire body. You heard it right. It controls your entire body. That's the master of you. What's the master of your body? Brain. So definitely we'll be studying about brain in this chapter. So let's get started with the basics first. So what is a nervous tissue? So we have already done the basics of the nervous tissue in the chapter structural organization in animals. So what is a nervous tissue? It is one of a kind of a tissue out of these four tissues that makes your entire body. So this is very important tissue, and it comprises of two types of cells. One are the neurons and other are neuroglial cells. Neuroglial cells are very small cells that are basically your nerve cells. What are these nerve cells, right? So neuroglial cells are basically nothing, but these are nerve cells. They are actually more than 50%, right? Okay, more than 50% in your nervous tissue are neuroglial cells. They are smaller than the neurons, whereas these are the structural and functional unit, and these are excitable cells. What is excitability? Excitability is a property where the cell can show excitation or the cell can conduct the electrical impulse. So you heard it right. Your neurons can conduct the electrical impulse. Yes, they can conduct the electrical impulse just like an electricity. So it just needs the movement of some ions across its membrane, right? So whenever the ions will move, they will give a charge to your neuronal membrane, and this is how your neuron conduct the nerve impulse or electrical impulse, and you call it as the cell is excitable. Any cell that can respond to this excitability property, the cell is also the excitable cell. For example, the neurons can conduct this electrical impulse, they're excitable cell. These electrical impulse can travel through muscles as well. We have done in the chapter body fluids as well as in locomotion. So the muscles, they are also excitable cell because they can respond to that electrical impulse. This is one property of neuron. This is one property of neuron and muscle that is, for both. That means the neurons and muscle. The neurons and muscle both are excitable cell. So we have done a very, beautiful diagram of a neuron in the chapter structural organization in animals. So let's see, how does a neuron looks like? So the body of a neuron is divided into two parts. Okay, so that's a revision for all those who have already done or attended my lecture, structural organization, and those who have not. Don't worry, you will study that with me here. Okay, so the entire structure of a neuron is divided into two parts. One is cyton, also known as cell body or perikaryon. All right, so this one, this is a cyton. You're thinking, ma'am, it has a name cell. So that means it will be having certain properties like of a cell. How many of you are thinking right now? I know number of you are thinking right now, right? So here it will be having a nucleus inside like this. So just like a normal cell, a cyton of your neuron will be having a nucleus. It will also be having organelles like Golgi bodies. These are Golgi bodies. These are what? Golgi bodies. All right. Golgi bodies. And then you can see these structure. What are these? These are dendrites. They are like arms. They are like arms. For example, you are a neuron, right? So if you're sitting alone, just do one thing. You're a neuron. Or if you are even sitting with your friend, You can ask your friend to do this. Imagine you're a neuron and you have these two hands, and ask your friend to clap on your hand, right? Or if they have to call you, for example, you are sitting like this. If your friend has to call you and you're just studying like that, so what that friend will do? Either they will call you by just speaking, or just they will say, hey, listen, right? But for example, you are having headphones on your ear, then you will not be able to hear, right? So what the friend will try, and what your friend will do, the friend will try to clap your hand. So by that clap, you get an impulse, and your that impulse goes to your brain, and brain will suddenly react. What's wrong with you? Like that, right? So just like that, these dendrites, they are also the one that receives the information, that receives the impulse from the another neuron. So your other friend was another neuron. Okay. So here, what are these structures known as? These are nothing but dendrites. Dendrites, as you can see, they are branched and they are afferent. Afferent means anything that will bring something. Just like in the kidneys, we have afferent arteriole that brings the blood. Afferent, wherever the word afferent is used, that means afferent means you're bringing something in. You're bringing something in. Okay. So they're branched, they are afferent, and they will receive the stimulus. They will receive the stimulus. Apart from that, a very important organelles are also present here. These are Nissen's body. What are these? These are Nissen's body. So these are Nissen's body, or Nissen's granules. Sometimes they're also known as Nissen's granule. They perform a very important function, because if they are not present, your neuron will not able to work properly. Yes. How? I will let you know soon. So Nissen's body, they basically contains ribosomes and RER. What is RER? Rough endoplasmic reticulum. And they will help in protein synthesis. That means if wherever or anywhere if ribosomes are present, their main function is to help in protein synthesis. So without any doubt, if Nissen's bodies have ribosome, what they will be doing? They will be producing protein. Okay. Now comes this portion, which is your axon. So in the axon, this axon, it ends here with the formation of these small bulb or swelling like structures. You call it as, all right, everything vanishes, no problem. You call them as axonal ends. What do you call them as? Axonal ends. Okay, so these swelling like structures, they're known as end bulbs, and the entire structure, this portion, is axonal ends. So the ends of axons, they end up with the swelling, bulb like swelling. You call them as axonal bulbs, axonal bulbs, or synaptic knobs, right? So this entire portion is cyton, and this is axon. Now these axon, this is because cell, and definitely it will be having a plasma membrane as well. It will be having a plasma membrane as well. So the plasma membrane of axon is known as axolemma. It is known as axolemma. So this cell can sometimes be wrapped by another cell. You heard it right. This cell can be wrapped by some other cell, and the name of the cell is Schwann cell. What's the name of this cell? Schwann cell. So your axon can be wrapped, not always, it can be or cannot be, right? It can be wrapped by certain cells, you call them as the Schwann cell. This Schwann cell have a very important function, that it secretes a phospholipid. It secretes a phospholipid that acts as an insulator. So these Schwann cell, how are they wrapping your axon? For example, this is your axon and this is your Schwann cell. So these Schwann cell will take up the axon like this and they will wrap it up. And inside that, it will be secreting that white color phospholipid, that is your myelin sheath. So yes, what's the name of this? That's a myelin sheath. Since myelin sheath is a phospholipid, so definitely it will act as an insulator. What is an insulator? The place from where no electricity can be passed, right? From where no electricity or electric current can be passed. Now you can see there are certain empty spaces. There are certain empty spaces like this one and this one where your myelin sheath is not present. There are certain spaces in between where myelin sheath is not present, or I can say the myelin sheath is absent. That place is known as node of Ranvier. Node of Ranvier. Right? So node of Ranvier is a place where myelin sheath is absent, and from here the electric current can be easily passed. Fine. So here let's talk about these bulbs now. So as these axonal ends have these swellings or bulbs, you call them as axonal bulbs or synaptic knobs. These synaptic knobs have small sac-like structure. These sac-like structures are known as synaptic vesicles. What are these? Synaptic vesicles. Right? These synaptic vesicles, they store the chemicals or the proteins that are formed by the Nissl's body. And this is how the Nissl's body are very much important. But now again, you don't know what's the function of these chemicals. So these synaptic vesicles, they are synaptic vesicle, these small sac-like structure. These contain some chemicals and they act as a neurotransmitter. What do they act as? Neurotransmitter. Just like in locomotion and movement, we have seen that these neurotransmitter are the one that helps in communication between a neuron and a muscle. So here the same thing will happen also, that these neurotransmitter, they will help in the communication between two cells. How these dendrites are taking information from the other cell? The cells are not clapping each other, right? So they are sending the information through one neuron to the another with the help of certain chemicals. And these chemicals, they are always produced in the cyton and they are released through the axonal ends. They are released through the axonal ends, right? So here, these small sac-like structures, they are synaptic vesicles. These are what? Synaptic vesicles. And the chemical inside them is a neurotransmitter. What are these? Neurotransmitter. These are nothing but chemicals. Let me give you an example. Do you know any example of a neurotransmitter? If you know, you can just say it because I can't hear it from now, right? From here. So the example is acetylcholine, the one which we have done in the muscles also. Okay. So for example, for an instance, if I say that there is another neuron just lying here, like this, they're forming a junction. For example, this is neuron 1 and this is neuron 2. Both are forming a junction and the name of a junction is synapse. What do you call it as? Synapse. So these chemicals will be released from the neuron 1 and they will attach to neuron 2 and then neuron 2 will accordingly respond. Okay. Also, this neuron can also end on muscles. What are these muscles? And you do not say the junction is a synapse. You call this as neuromuscular junction. What do you call it as? Neuromuscular junction. Neuromuscular junction. Fine. So this is how one neuron communicates with the other. We'll also be discussing this in the next sections in the detail. So before that, let's talk about what are the various types of a neuron. Now on the basis of polarity. Polarity means what we say. What is a polarity? Like on Earth, we say there is a North Pole and there is a South Pole. So that's a polarity. Just like that, neurons, they also have polarity. How? For example, if I say this is one pole and this is the another pole. In this, you can say on the first pole, this one, there are a lot of dendrites present. And on this one, only one axon is present, though it's bifurcating or trifurcating, but this is only one axon. When a neuron, for example, have a lot of polarity on one side, you call it as multipolar neuron. Okay. For example, if I say I have a neuron, with this cyton and only one axon and no dendrite. So it has only one polarity. Dendrite and axons are basically making it bipolar, right? For example, in case of bipolar, if this is cyton, this is a dendrite and this is the axon. So it has two poles. Right, so it is a bipolar. So if I have a neuron with a lot of dendrite, it's called multipolar with one axon. If I have a neuron which was formed as a unipolar like this, but later on it changes its mood and it decided to form one axon and one dendrite. This is considered as pseudo. Pseudo means false, pseudo unipolar. Pseudo means false, unipolar. That means it's a false unipolar. This is a liar, 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 pants on fire, right? Okay. So this one is unipolar. In unipolar, the one thing that you can see, there is only one axon. In the bipolar, what you can see, in bipolar, there is a one axon and one dendrite. Whereas in the multipolar, you can see number of dendrites. Whereas only one axon. Fine. So there are different types of neurons on the basis of this structure. You can see that. All right. Now, unipolar, bipolar, multipolar, pseudo-unipolar. The question that are usually asked in the exam is what is the location of these neurons? Fine. For example, unipolar, they are not present in your body right now. They were present when you were an embryo. So unipolar are always found in the embryonic stages. Fine. Bipolar. Bipolar is present in the retina of eye. It's also present in the olfactory epithelium. So where have we studied olfactory epithelium? Where have we learned about the olfactory epithelium? We have learned it on in that chapter, breathing and exchange of gases, where I made you understand the structure of the nose, the inside regions of the nose, and there is one olfactory epithelium in the olfactory region of your nose. Fine, guys? Okay. Multipolar. Multipolar is present in your CNS. What is CNS? Central nervous system. So I'll also be explaining you different types of nervous system, and central nervous system comprise of brain and spinal cord. You all know what's a spinal cord? Yes, it's a long rod-like structure present inside your backbone. Okay, and it emerges from brain. Next, pseudo-unipolar. The pseudo-unipolar is found in the dorsal root of spinal cord. Now what's that? If I take the section of a spinal cord, this will be the dorsal side of spinal cord and this will be the ventral side of spinal cord. So on the dorsal side, the nerves are emerging out like that, right? So because spinal cord is a part of your central nervous system. So from here the nerves are emerging out, and here you can see the pseudo-unipolar neurons are present on the dorsal root of your spinal cord. Okay, and these are the various types of your neuron. Let's talk about various types of nerve fibers. So nerve fibers and neurons, they are, sometimes they are used as synonymous also. What basically are nerve fibers? So whenever I'm talking about the nerve fiber, I'm talking about the long axons that are spread across my body. Okay, so whenever I'm talking about the nerve fiber, I'm talking about the neuron only. But what part of neuron? The long axons of my PNS. What is PNS? Peripheral nervous system. What is peripheral nervous system? I'll be talking about that shortly. Okay, so these nerve fibers, they are of two types. One is unmyelinated and another is myelinated. Okay, so the myelinated nerve fiber, from the name it suggests, the myelinated have myelin sheath. They have myelin sheath. The myelin sheath is present. You can find them in the cranial and spinal nerves. You can find that in cranial and spinal nerves. What are cranial nerve? The cranium word is always used for brain. So these are the nerves of brain and these are the nerves of spinal cord. Okay, whereas in unmyelinated, myelin sheath is absent. Now there is a point. Whenever I'm saying unmyelinated, whenever I'm saying myelin sheath is absent, in unmyelinated nerve fibers, maybe Schwann cells can be present, but myelin sheath will be absent. There can be a possibility that a Schwann cell is wrapping an axon, but maybe that Schwann cell is not producing any myelin sheath. Fine. So here myelin sheath is absent, Schwann cell may or may not be present. So where do you find these one? In the autonomic nervous system and somatic nervous system. So enough with the types of nervous system. Now it's high time that we should learn about the various types of nervous system. Okay. So but before that, I will also explain you another classification of nerve fibers. Another classification of nerve fibers. Nerve fibers can also be of two types. One is afferent, another is efferent. As the name suggests, afferent is bringing something and efferent is taking away something, right? So afferent are the nerve fibers that will give information to central nervous system, whereas efferent is the one that will take away the information from central nervous system. That means it will give information to the brain, this will take away the information from the brain, right? So these are sometimes also known as sensory nerve fibers, and they are sometimes known as motor nerve fibers. Fine. For example, there is any sensory receptor. For example, you touched something very hot. So on your skin, you touch something very hot. Now what will happen? Imagine I touch something hot. Oh, I did it like that. That's a reflex. Okay. So what happened at this point? My skin have sensory receptors. Through neuron, they are giving information to my brain or my spinal cord, my central nervous system, right? Now, these nerve fibers that have sent information to my central nervous system, they are afferent. So they are sending information to CNS. Sending information to CNS. Whereas, now what will happen? I have to take away my arm. So my muscle should contract. So at this point, my central nervous system will send information to my muscles. And that means the information is going away from the central nervous system. They take away the information from the central nervous system. So on the basis of where the information is going on the basis of what we have taken, central nervous system, from where the central nervous system is sending information, or we can say how the information is going across, it's going towards the CNS or away from the CNS. We have divided the nerve fibers into two, the afferent and efferent. Now enough with the this portion, that is your neurons and nerve fiber. Let's talk about the nervous systems now. So you might be confused, ma'am, I don't know what's autonomous. Ma'am, I don't know what is PNS. Ma'am, I don't know what is CNS. Help, help, help. No problem. We are here to discuss neural system. So neural system is of two types. One is central nervous system, which is CNS. Okay. And other is PNS, peripheral neural system. Peripheral neural system. You can also write it as nervous system. You can also write it as neural system, one and the same thing. Okay, neural or nervous system. Now guys, what's the difference between two? So peripheral, so the central nervous system, it comprises of brain and spinal cord. It comprises of brain and spinal cord. So brain and spinal cord, they are collectively known as central nervous system. So if this is your brain and this is your spinal cord, this is your central nervous system. Now all the nerves that are emerging, from your CNS, that forms your PNS, peripheral nervous system, right? So what is peripheral nervous system? All the nerves that emerges from CNS, that forms your PNS. Now, out of these nerves, there will be some nerves that are sending, imagine these are certain nerves, they are sending information to your skeletal muscle. Okay? And there can be certain nerves like these nerves or these total nerves, they are sending information to visceral organs. Okay, so according to where these nerves are sending information, we have divided our peripheral nervous system into two categories. How many categories? Two. One we have is somatic neural system. One we have is a somatic neural system. Another we have is the autonomous neural system. Okay, so on the basis of where these nerve fibers are innervating, where are they sending the information, we have divided our peripheral nervous system into two portions. One is somatic nervous system. In the somatic nervous system, these nerves are sending information to the skeletal muscles, right? So here skeletal muscles are involved. Whereas in autonomous, the information is sent to the visceral organ, so that's why it is also known as visceral nervous system. So it is of two kinds further. One is sympathetic and another is parasympathetic. One is sympathetic, another is parasympathetic, right? This one is little complicated because it contains a lot of ganglion, right? It's, its entire structure is little complicated because it is innervating your essential organs. For example, your heart. In the heart, we have done in the regulation part of the heart that your heart's heartbeat can either be increased or it can be decreased. So it is under the control of your cardiovascular central of your medulla, which will send information through neurons. If these neurons are of sympathetic nervous system, they will increase the heartbeat. If they are of parasympathetic, they will decrease the heartbeat. So as you can see, both are working opposite to each other. So always sympathetic and parasympathetic, they work opposite to each other. So sympathetic and parasympathetic, they always work opposite to each other. So this autonomic nervous system, it includes a lot of nerves, ganglion and visceral organs, obviously, etc. Fine. This entire structure we'll be discussing in detail when we'll be discussing about the autonomic nervous system in detail. Okay. All right. So that's about your entire classification of nervous system, guys. Let's read the lines of NCERT because I don't want you guys to miss this portion. So that's the entire structure of your neuron. We have already made one. I don't think so any of the things are left. These things might confuse you, but you don't need to. Axon terminals are also known as axonal ends that we were talking about. Okay. These are end bulbs. This portion is axonal terminal. Okay. All right, so we'll start from these. The axon is long fiber, the distal end of which is branched. Each branch terminates as a bulb-like structure called synaptic knob, which possesses synaptic vesicle containing chemicals called neurotransmitter. So every end bulb have vesicles known as synaptic vesicle. Inside them are present chemicals known as neurotransmitter. The axon transmit nerve impulse away from the cell body to a synapse or to a neuromuscular junction. So where it is sending information, either to a synapse, that means to the another neuron, or to the muscle through neuromuscular junction. Based on the number of axon and dendrite, the neurons are divided into three types. So how many axon and dendrites are there? We have divided accordingly. Multipolar with one axon or two or more dendrite found in cerebral cortex. Now what is a cerebral cortex? We have written multipolar are present in CNS, and CNS comprise of brain and spinal cord. The brain's inner area is known as cerebral cortex, right? So we have written here an example that it is present in the CNS, brain and spinal cord. So in brain, inside the brain, we have an area known as cerebral cortex. So they are present in that. Okay. All right. Next. Bipolar dendrite found in the retina of the eye and also olfactory epithelium. Unipolar, they are present in the embryonic stages. They have only one axon. They have not mentioned about pseudo-unipolar, but I hope you will understand, some things we need to do out of the NCERT also. There are two types of axon, namely myelinated and non-myelinated. The myelinated nerve fibers are enveloped with Schwann cell, which form a myelin sheath around the axon. So in the myelinated one, the myelin sheath is present. The gap between two adjacent myelin sheaths is called node of Ranvier. Myelinated nerve fibers are found in spinal and cranial nerve. Unmyelinated nerve fibers are enclosed by Schwann cell that does not form a myelin sheath. As I have told you, the only difference between myelinated and unmyelinated is on the basis of myelin sheath, not on the basis of Schwann cell. In the unmyelinated, Schwann cell may or may not be present. and is commonly found in autonomous and somatic neural systems. Fine. Okay, let's read the NCERT of this also, and then we will solve the questions. The human neural system is divided into two parts: the central nervous system or neural system and the peripheral one. The CNS include brain and spinal cord and is the site of information processing and control. As I've told you, who is controlling your body? The brain. So it's the site where everything is controlled. The PNS comprise of all the nerves of the body associated with the with the CNS. So all the nerves that emerges out from CNS, they form PNS. The nerve fibers are of two types, afferent and efferent. Afferent are sensory, efferent are motor. Afferent are nerve fiber transmit impulse from the tissues or organs to the CNS, and efferent transmit regulatory impulses from the CNS to the peripheral tissues or the organs. Okay. The PNS is divided into two divisions: somatic neural system and autonomic. The somatic neural system relays impulses from CNS to skeletal muscle, while the autonomic neural system transmit impulses from CNS to the involuntary organs and the smooth muscles of the body. So as I've told you, it goes to the visceral organs. So it's quite obvious if I'm talking about visceral organs, that means I'm talking about the smooth muscles also. I'm also talking about the involuntary organ, because visceral muscles are involuntary. Is your gut under your control? No. Is the lungs under your control? No. So they are going to your involuntary organs. Okay. The autonomic neural system is further divided into sympathetic and parasympathetic. Visceral nervous system is the part of peripheral nervous system that comprise the whole complex of nerve, fiber, ganglion and plexus by which impulse travel from the central nervous system to the viscera, from the viscera to the central nervous system. So as I've told you, the visceral nervous system is also known as the autonomic nervous system, and it contains your fibers, ganglion and plexus. What is a plexus? These are the collection of nerves or neurons, collection of cell bodies, or we can say web, not the collection, synapses or web of cell bodies. For example, this is one cell body, this is a dendrite, this is the another cell body. So this is how they are forming a web network like, right? Web or network like structure known as plexus. Otherwise, we'll be doing this in detail in the later section, so don't worry about that. Okay, that's about the NCERT, guys. That's about the NCERT. Let's solve the question. The nerve fiber that transmit impulse from tissues or organs to the CNS are. That means from tissues, there is a T, okay. From tissues, the information is going to CNS. Towards the CNS, that means we are talking about afferent. Answer is 1. Okay, next. The dash neural system relays impulses from the CNS to skeletal muscle. That is voluntary muscle. That's a part of your peripheral nervous system, and you call it as somatic neural system. Answer is 4. Next. The neuron with one axon and one dendrite are found in. That means they're talking about bipolar. So where do you find the bipolar? In the brain? No, in the brain, which one do we have in the brain? We have multipolar. In the retina, we do have bipolar. In dorsal root ganglion, we have pseudo-unipolar, whereas in embryo, we have unipolar. So answer to this question will be second. Next. Nerve impulse. So now we are going to start with the very, very important topic that you call it as a nerve impulse. So let's get started. So let's get started with the topic that is nerve impulse. So you all must have heard of nerve impulse, right? Or impulse. It's not that pulse that you detected here through the wrist, and neither this is the pulse toffee that you eat. Yeah, there is a candy named Pulse also. So nerve impulse, what is it? We say that neurons are excitable cells. What are excitable cells? That can generate the electric current, or they can also detect the electric current and they can respond to that. Right. So nerve impulse is an electrobiochemical phenomenon. It's just like an electricity or an electric current that is flowing across the neuron's membrane, right? So what is a nerve impulse? We say that our neurons are excitable cells. So it's basically the electrical signal. It's what? It's the electrical signal that transmits or flows or travels through a nerve membrane. Through a nerve membrane. So this is what excitable means, that any cell is excitable if it can detect or it can generate this electric current, right? So we say that if this is your neuronal cell, if I say it is a cell membrane, it's a neuronal cell membrane. It's a neuronal cell membrane. We say that a current is flowing through this, neuronal cell membrane. Then why or how does this electric current moves, and what is that phenomenon that leads to the formation of this current? So there is one thing that are responsible for generation of this current is a stimulus. So whenever a stimulus arises, the nerve cells gets excited. They started producing electric current. So now, yes, okay, fine. The stimulus arise and they got excited. Now, what is the thing that leads to the change in charge? So that are the ions. So this cell membrane leads the flow of ions. So for the flow of ions, we need a passage, because the cell membrane is made up of lipid. If I'm saying only if the ions are moving across the membrane, the charge developed, and that leads to the formation of electrical signal. So if I say the membrane is lipid and lipids are insulator, they will not allow the formation of this charge. Then how will the charge will be formed? So for that we need we have certain ion channels. We have certain ion channels. For example, this is sodium ion channel. So when this open, that leads to the movement of sodium ions. Right? So what are these? These are ion channels. So ion channels are nothing but these are just proteins. These are the doors made up of protein across the cell membrane, and that will allow the movement of the ions, leading to the change in the charge and hence the voltage. And then what we say, not in fact the voltage, you always detect the electric current in millivolts, just like that. So that will lead to the formation of a kind of an electrical signal. Now these ion channels, they are of different types. These ion channels, they are of different types. First of all, what are these? These are made up of protein, and they allow the passage of ions. Allow the passage of ions. Fine. So now what happened here, guys? Ion channels, they are of different kinds depending upon what type of ions they are transporting. For example, this one is only helping in the transport of sodium ions. So this is a sodium ion channel, right? So there are different kinds of channels. Some channels are leaky channels. They are open all the time. Right? They can open, they can close, just like this one. These are normal ion channels like sodium ion channel. So sodium ion channel will only be leading to the transport of sodium ions, and potassium ion channel will only lead to the the transport of the potassium ions. Then we have certain channels which can only open and close according to the stimulus. Right? If that thing will come, only then they will open. For example, if I say there are ligand gated ion channels. So ligand gated ion channels need to have certain chemicals that will open them. For example, if I say this is the sodium ion channel, but this is not the leaky one, this is not a normal sodium ion channel, this is the one which is a ligand gated. For example, if to this ion ACH, what is ACH? A neurotransmitter. If to this channel the ACH binds, only then this will open and lead to the transport of sodium ions. So they will open and close according to the chemical that binds. According to the chemical that binds. Right? And then we have the voltage gated ion channels. Voltage gated. Now what are these? If ever there is a change in the potential difference that is same you study in the physics, right? So if there is any change in the potential difference across the cell membrane of neuron, only then these ion channels will open. So they open and close with respect to WRT, Diksha ma'am always write WRT, that is with respect to, right? With respect to the change in potential difference. Potential difference. Fine? So like we have sodium ion voltage gated channel. So as you can see, what is that common thing? The common thing is everyone is transporting sodium. There can be sodium ion channels in the leaky, there can be sodium ion channels in the ligand, there can be sodium ion channels in the voltage. The only difference is what is the kind of stimulus that open and close it. For example, for opening this kind of a channel, you need to have a ligand, a chemical. To open this kind of a channel, you need change in the potential difference. To open this kind, they open and close randomly. You do not need a lot of stimulus for that, right? And sometimes, yes, stimulus is needed, but normally they are always in the open state. Fine? All right. So that's about the ion channels that leads to the transport of ions and which lead to change in the potential difference, change in the charge, and hence that will lead to the production of electric current. Now this is about the theory. Now how actually the generation of impulse takes place. So for that you need to understand one very important topic, that is, okay, RMP. What is RMP? Resting membrane potential. What's the full form of RMP? Guys, resting membrane potential. Resting membrane potential. Now what is this? So whenever our neuron is resting, no stimulus have been arised, and it is not generating any impulse. At that time, the situation in which the neuron is sitting silently doing nothing is known as resting membrane potential. And you know, you must have thought, ma'am, but that's a potential. Very true. The resting membrane potential is the potential of your nerve membrane when it is at resting state. Again, I'm telling you, RMP is resting membrane potential. It's the potential difference across the cell membrane. What cell? Neuron. When it is doing nothing, when it is just resting, right? So what is resting membrane potential? It is the potential. It is the potential across the neuronal membrane when it is at rest. When it is not doing anything, it is completely at rest, right? So let's see what is that. For example, again, this is your neuronal cell membrane, and if I say this is the outside and this is the inside. So we are not drawing the entire cell. I have just drawn one axonal membrane or membrane, cell membrane of axon, right? So we say that outside always, that means outside the cell, always the concentration of sodium ions is higher, and inside always potassium ions are higher, right? So we always say that whenever we are talking about a cell, outside the concentration of sodium ions, that in your body fluids and in the interstitial fluid. What is an interstitial fluid? The fluid present in between the tissues, right? And the potassium ions, they are higher inside the cell. Okay. Now, what happened during resting state? Our entire membrane is more negatively inside and positively outside. We say that during resting state, our cell membrane have a certain charge difference. Inside is more negative and outside is more positive. As a result, across the membrane, if you will see, the difference in the voltage, or we call it as difference, or you call it as potential difference, then inside it is more negative. So you call it as, it is how much negative? Inside is minus 70 millivolt. Inside is minus 70 millivolt. That's the potential difference across the cell membrane. So we say inside it is more negative, outside it is more positive. Now, what are the factors that lead to this minus 70 millivolt situation? Why it is always minus 70 millivolt? Or why we say that sodium concentration is higher outside and potassium is higher inside? Why so? Why so this happen? There are three reasons. I say sodium is always higher outside, potassium is always higher inside, and the potential difference across the cell membrane is minus 70 millivolt, right? Why is it happening? Why is it happening? There are 3 reasons. How many reasons, guys? 3 reasons. Let's check them out. First reason is differential permeability. Differential permeability of ions. What does it mean? Differential permeability means there is a difference in the permeability. Permeability means anything that is allowed to move or not. For example, if I say sodium ion can run faster, potassium ion runs slower across the membrane, then we will say they have different types of permeability. Permeability, that means allowance to move. Fine. So why does this happen? For example, if I say this is a cell membrane, it has more number of potassium ion channels than sodium ion channel. If I say there are more number of potassium ion channels than sodium ion channels, then what will happen? Just think, have some brainstorming. Just use it, otherwise it will go bad, you know. So if I say there are more potassium ion channels than sodium ion channels, then what will happen? And we always say sodium is higher outside and potassium is higher inside. And you have also studied somewhere in the botany that things always move from high partial pressure to low partial pressure, or we say in case of osmosis, it always moves from high concentration to low concentration. In zoology, you always study high partial pressure to low partial pressure, and in botany, according to osmosis, the things always move from high concentration to low concentration. So how will these things move? If I say this is one sodium ion channel, only one sodium ion will enter, right? Whereas more potassium ions will move outside. So as you can see here, 2 potassium ions have move outside. So how many positive charge the outer part of a membrane have? 2. Now only 1 sodium ion have entered inside. Now how much charge the inside membrane have? 1. So if we will create a difference, the inside one is at loss or at negative side of 1 as comparison to outside. So we say that outside is positive, inside is negative. Why? Because there is more efflux. More efflux. Efflux means moving outside. Efflux means moving outside. More efflux of potassium ions than influx of sodium ions. As a result, the outside is more positive and inside is more negative. This is the reason why outside is positive and inside is more negative. This is the first reason. Okay, now let's look about the second reason. The second reason is sodium potassium ATPase pump. Sodium potassium ATPase pump. What are pumps? These are not the hand pumps that will pump water from the groundwater and give it to you. No. It's not like that. Pumps are what? For example, if this is the membrane, in the membrane, certain proteins are present. What are present? Certain proteins are present. These proteins, they act as ATPase. How do they act as? ATPase. And they have seats. You heard it right? Seat to sit on. And these seats are not for you to sit on. These seats are for the ions to sit on. Now, what it will do? It will pump 3 sodium ions outside and 2 potassium ions inside by using 1 ATP molecule. You know, this ATP with the help of this enzyme will be converted into ADP and IP. And you all know ATP is the energy currency. When IP is lost, it will give energy and it will lead to the functioning of the pump. And what will pump do? Pump will, for example, if this is a pump, okay, this is the inside, this is the outside. This is the inside, or you just take it this manner. This is the outside, this is inside. On this side, which is facing towards me, the 3 sodium ions will sit here and 2 potassium ions will sit here. Now ATP will come the energy and this pump will move like this. As a result, the 3 sodium ions will be pumped outside and 2 potassium ions will be taken inside. So there is a benefit again. 3 sodium ions are moving outside. 1, 2, 3. Only 2 potassium ions have been put inside. So again, the inside is more negative, outside is more positive. So this is outside and this is inside. Fine. Another thing, you also have a benefit of something. We say that the concentration of sodium ion is always higher outside. So these pumps will make sure the sodium ions should be higher outside and potassium ions should be higher inside, right? Now the third reason, let me just use another page for that. Okay, what's the third reasons? The anions. What are anions? Anions are negatively charged. They are negatively charged and they are larger in size. They are larger in size than the cations like sodium ion and potassium ion. Now what happened? Imagine this is the neuron. Okay, and this is the membrane of neuron. And we say that in the membrane outside there is always positive charge and inside there is negative charge. So one such reason is differential permeability, second the pump, and third anions. Anions do not have the channels to move, right? Anions are trapped inside your cell. Anions are trapped inside your cells and they do not have any channels to move outside. So what happens whenever there is accumulation of positive charge, whenever there is accumulation of positive charge to the outside, they will get attracted and they will accumulate towards the membrane like that. Anions are always trapped for just like a magnet. Like for example, if you're sitting in a room and I give the magnetic charge to the entire walls and you put some iron thing inside, or if I say I give charge or I make your walls like a magnet and I put a one iron thing inside the room, then when that magnet or when the walls are converted into magnet, that iron thing will get attracted toward the wall. Just like that. So there also, when the positive charge accumulate outside, the anions, they will move towards the membrane. As a result, there is a creation of the charge difference. The charge difference has been created along the membrane and hence inside is more negative and outside is positive, right? So we say that the anions, we say that these anions are negatively charged. They do not, let me just write it with another color, they do not have any anion channels for their movement. So they get attracted towards positively charged anions present outside the membrane, creating a charge difference, creating a charge difference across a membrane, across the membrane. So this is how there is a negative charge towards inside and positive outside. Let's see what NCERT has to say. Okay. So neurons are excitable cells because, okay, that might not be clear to you. Let's increase the font. Okay, that's my way of teaching, guys. I always use NCERT clips so that you will not face difficulty later on, because there are certain sentences in the NCERT that are way difficult for you guys. So that's my habit to give the NCERT. Okay, so those who are there for the first time, who have not seen any of my lectures before, they must be knowing that. But those who are new here, you're a new student, and so that's the way how your ma'am teaches, right? Okay. So do you know why the membrane of a neuron is polarized? So as I said, there is a charge difference. You can also call it as polarized. Different types of ion channels are present on the neuronal membrane. As I've told you, there are ion channels present. These ion channels are selectively permeable to different ions. So selectively permeability means differential permeability means different types of permeability. So because they're selecting things, so you can also call it as selective permeability. Okay. When a neuron is not conducting any impulse, that is resting, the axonal membrane is comparatively more permeable to potassium ions and nearly impermeable to sodium ions. This is what we have done. So whenever it is at resting membrane, potential is there is differential permeability, there are more number of potassium ions, that means there is more efflux of potassium ion, and this is what you call it as permeability also. Then similarly, the membrane is impermeable to negatively charged proteins present in the exoplasm. Consequently, the exoplasm inside the axon contain high concentration of potassium ion and negatively charged proteins and low concentration of the sodium ion. So we say that here that the anions, what are these anions? They are basically present in the form of proteins. So anions, what anions can be there in the cytoplasm? They all are present in the form of what? Proteins. These are basically what? These are basically all proteins, right? Okay, so as a result, they are always present towards inside, and they in the next line they have said that inside the potassium ions are higher and outside sodium ions are higher. All right. In contrast, the fluid outside the axon contain a low concentration of potassium ion, a higher concentration of sodium ion, and thus form a concentration gradient. So there is a gradient form. That means we always say sodium is higher outside, potassium higher inside. These ionic gradient across the resting membrane are maintained by the active transport of ions by the sodium-potassium pump, which transport 3 sodium outward and 2 potassium inside the cell. So here we have discussed, there I have told you also that whenever this pump works, it solves 2 purposes. One, it will definitely create negative charge inside. Second, it will make sure that sodium ion should be higher outside and potassium ions should be higher inside, right? Okay, next. All right, while its inner surface becomes negatively charged, therefore is polarized. The electrical potential difference across the resting membrane is called the resting potential, right? So this is all about why you have minus 70 millivolt, a polarized state, or you can say during the resting membrane, when the membrane of your neuronal cell membrane is completely in a rest form. And as I've told you, we are always talking about this membrane is a membrane of the neuron. And what part of a neuron? Axon, right? So it's basically what? Axon. So this cell membrane is always of neuron. You must write it. Why? Because you know, after some time you'll get confused. Now you are hearing everything and you must be thinking, yes ma'am, okay, done. But always make good notes, guys. Okay, so here the cell membrane, and this cell membrane is of a neuron and basically of axon. So you also call it as exoplasm. What you call it as? Exoplasm, or sorry, you call it as exolemma. Exolemma, just cut the N here. Exolemma. Exolemma is a cell membrane, whereas exoplasm is a cytoplasm. Alright, so that's all about why there is a resting membrane potential. Now let's move further and talk about how the nerve impulse is generated. Let's see. Now guys, for example, this is a membrane and we say positive outside, negative inside, and this is RMP, and at RMP we have minus 70 millivolt. Okay, and we say this is outside, this is inside. Outside always the sodium concentration is higher, and inside always potassium concentration is higher. This is what we do. Okay, now moving further. So what we say here is that how does this electrical impulse is generated? First of all, always there is you know, a stimulus that arises. Always, how does the neurons respond? What do you think? How does the neurons respond? You know, for example, right now, if someone touches you, you are sitting in a table and someone touches you and asks you to talk to them, they will touch you, okay? Just like any of your friend is sitting behind you. So how that friend will call you? the friend will touch you, like they touches you here, okay, and now suddenly you will move and say, what's the problem? Right? So just like that, that touching was a stimulus. So stimulus is something that is change in the environment, right? Stimulus is something that is change in the environment, okay? So whenever the stimulus arises, there is a change in environment that leads to opening of sodium ion channels. Which ion channels open here, guys? Sodium ion channels. Now, if the sodium ion channels open and concentration of sodium is higher outside, sodium ions will enter inside. So the stimulus causes opening of sodium ion channels. Okay, so when the sodium ion channels open, the sodium ions are moving inside. One confusion that you always have, ma'am, but the positive charge is moving, then there should be, negative charge. No, the ions are moving, the electrons are not moving. Okay, so when the sodium ions move inside, the inside of a membrane will start it to become positively charged. As a result, from minus 55, it will first move to minus 55 millivolt. And this is a kind of threshold stimulus. What is a threshold stimulus? That this is a minimum stimulus that you need. This is a minimum stimulus that you need to reverse the polarity. This is a minimum stimulus that you need to reverse the polarity. For example, the sodium ions are entering. Now, as a result, one situation will come where the polarity got reversed. So, once it has entered the minus 55 state, it will definitely go to this stage, which is of plus 30 millivolt. So that's why you call it as threshold stimulus. For example, if your membrane comes to around minus 40 millivolt, okay, it comes to around minus 40 millivolt and it stops there. So that means from there it will not go to plus 30 millivolt. Okay, why? Because that's not the threshold stimulus. We need minimum minus 55 millivolt to convert it into plus 30 millivolt, which is the entire opposite to it. That means we are reversing the polarity. And this stage from RMP to plus 30 millivolt, this one where polarity has been reversed, you call it as depolarization. Depolarization. And here you say the action has been done, so that is known as action potential. What do you call it as? Action potential. So now plus 30 millivolt have been, right, the membrane has become excited. This is why you call it as action potential. The action has been done, and what is that action? That means your membrane has got excited. Okay, now what will happen next? Now this will lead to the opening of potassium ion channels. And potassiums are higher inside, so they will move outside. So now there is opening of, due to plus 30 millivolt, there is opening of potassium ion channels. Now once the potassium ion channels open, again the reversibility will occur. Inside will become negative, outside will become positive. And you call it as repolarization. You call it as repolarization, and here the potential difference is minus 70 millivolt. Now you must be wondering, ma'am, this is also minus 70, ma'am, this is also minus 70, then why do we call it as our resting membrane potential? Why are we calling it as repolarization? We can also call it as from RMP go to depolarization, then come back to RMP. Why this is not called as RMP? It is known as repolarization. The reason is, here the ionic concentration gradient has also been disturbed. Here the ionic concentration gradient has also been disturbed. During repolarization, sodium ions are higher inside and potassium ions are higher outside. Yes, yes. Why? Because potassium have gone outside and sodium during depolarization, they have entered inside. Now how will we correct it? Who is the one that can correct this? Who is the one that can throw 3 sodium ions outside and take up the 2 positive ions inside? That is your pump. That is your pump. So this pump will use one ATP, throw 3 sodium ions outside, put 2 potassium ions inside and get it back to minus 70 millivolt, right? And that will be your RMP, that is minus 70 millivolt. Okay, and this is how we get back here. Sometimes from minus 70, your this charge can also go to more negative, that is around minus 90 millivolt. Sometime if potassium ions have been opened for longer duration, they forget we have to move back. So potassium ion channels, they forget that we have to move back. So they're like open for long time interval. So that time, this stage is known as hyperpolarization. That stage is known as hyperpolarization. Even if it has entered into the hyperpolarization, it can get back to resting membrane potential by the similar manner. That means the use of ATPase, sodium potassium ATPase pump. So this is how the excitability is generated. When the stimulus arises, sodium ion channels open, that will reverse the polarity and you say this is now excitable. How it will move back? It will then lead to the opening of potassium ion channels, which will lead to repolarization, and then the pump will work and that will go to resting membrane potential, and this is how it go back to rest, right? So this is the position when there is a stimulus that arises on only this segment of neuron. For example, your friend touched you only here. So that means the stimulus was only here. Then how did your entire body with the uses of your skeletal muscles move like this? How does the impulse reach to your brain and then from brain to your muscles, and how did muscles have taken up this impulse? Now that you call it as, my dear students, you call it as conduction of nerve impulse. What do you call it as? Conduction. See, one is generation of nerve impulse. That how with the help of stimulus at that one particular position, the resting membrane potential have been reversed and have caused depolarization. Now how in that segment where the depolarization has taken place by stimulus will move to the entire body. Now that is conduction. Okay, or we can say how, for example, if I say this is one neuron, okay, this is neuron. Imagine the stimulus have arrived here, then how does it will move towards the entire neuron? That is your conduction. This is your conduction. Right? Now, this neuron will end at these nopes. Okay. Now, how it will move from one neuron to another, and this you call it as transmission. Transmission. Okay. So where stimulus arises, here you call it as generation of nerve impulse. After generation, how that depolarization wave moves towards the exolemma of the entire neuronal membrane, that is conduction, and how from one neuron it goes to another, that is transmission. So now we are going to talk about the conduction. Now for example, if the stimulus arises here, at this point, the A segment, we have divided the entire axonal membrane into three segments, A, B and C. Now if the stimulus arises here, this will lead to the second step. In the first step, the stimulus arises. In the second step, what happened? The A segment, as you can see, it has undergone from minus 70 millivolt, it has entered to plus 30 millivolt, that is depolarization. So we say after stimulus, the A segment will undergo depolarization, right? Now, what will happen next? Now, what will happen here? There is a charge difference. As you can see, this is negative, positive, positive, negative. This charge difference will create a local circuit, and this local circuit will act as a stimulus for B segment, right? Because for depolarization, definitely you need the stimulus. The first one was a different stimulus, like your friend touched you. How the second segment will take up the stimulus? It will generate its own stimulus. Now for that, what's the stimulus? The charge difference, the circuit that has been formed here. As a result, the B, okay, as a result, the B will now undergo depolarization. Okay, and this one in meantime is undergoing repolarization. Now again you can see there is a charge difference, right? And now that will act as a stimulus for the C segment, and now C will undergo depolarization. And this is how the nerve impulse will get conducted from one site of stimulus to the entire exolemma, right? Okay, now conduction is of two types. One is continuous, another is saltatory. Continuous is the one that we have seen right now. It is just moving continuously because there are no barriers. right? There are no barriers or there are not interruptions. There are not any interruptions. That's why it is moving on continuously in a smooth manner. Whereas in the saltatory, for example, if you are moving, okay, and there are certain big rocks that are present, or you can say the rocks of your this 1 feet tall, they are present on some places. Then what will you do? You will step up, jump, and then move further like that. Okay, so if you have no rocks in your path, then you can easily move like that continuously, just like that. Okay, so this is what happened in the continuous conduction. In continuous conduction, they are usually occurred in the unmyelinated nerve fibers. In unmyelinated nerve fibers, there is no myelin sheath. For example, this is how the nerve fiber is. There is no myelin sheath. There is no myelin sheath. So the impulse will move in the manner that we have just done right now in the ABCD segment form. Fine. Whereas in the saltatory, what happened? There is a nerve membrane and there is the myelin sheath in places. So, because there is myelin sheath, it acts as an insulator. It will not allow the current to pass through. So, the current will move by jumping like this. That's why its name is saltatory conduction. Saltation means jumping, right? So, here nerve impulse jumps. So, what do you think? Which one is faster? This one is faster. Why? Because it is jumping and it will also use less energy. Why? Because only few segments have been depolarized. So you don't need to use a lot of pumps. More pumps you use, more there will be, ATP used, because pump always use ATP. For example, if I say there 1 ATP will be used, for this also 1 ATP will be used, for this segment 1 ATP will be used. Because every segment is getting repolarized, for reaching RMP, they will be using pump. Fine. But here, if on the B segment we have myelin sheath, then now how many ATPs will be used? 1 and 2. So less ATP will be used in saltatory conduction. Less ATP will be used in saltatory conduction. Okay, so this is how it can be conducting throughout the exolemma. Moving further, how does it is transmitted from one neuron to another? First of all, for transmission, we have synapses. What do we have, guys? Synapses. As we have already done here, synapses are of two types. One is a chemical synapse and another is the electrical synapse. Now, what's the difference? All right. So this is a very important topic because from here the questions have been asked in the previous years. So you don't skip this one and pay attention here. Okay. So what happened, guys? For example, you have this one neuron. This is another neuron. And you need to send impulse from this neuron to this one. And this junction between the two is synapse. The junction between the two is synapse. Okay, now this neuron, which is present before the formation of synapse, is presynaptic neuron. This is presynaptic neuron. And this one is postsynaptic neuron. This is postsynaptic neuron. Similarly, this membrane will be known as presynaptic membrane. And this will be known as postsynaptic membrane. Okay. Now, the first neuron have nerve impulse that rises to this position in the form of the waves of depolarization and repolarization. Imagine here the nerve impulse have been entered. All right, here nerve impulse have been entered and we need to excite this one or inhibit this one, or we can say we have to send the information from this neuron to this one, from presynaptic to postsynaptic neuron. Now what will happen? So as you know, guys, here some small vesicles are present known as synaptic vesicles. So when the nerve impulse arises here, there is opening of calcium ion channels. There is opening of calcium ion channels in the presynaptic membrane. So in the presynaptic membrane, due to entry of the nerve impulse, there is opening of calcium ion channels and calcium ions will enter inside. And this will lead to fusion of the synaptic vesicle with the membrane, and this will release the neurotransmitter. What happened? When the nerve impulse arises, the calcium ions will enter inside the neuronal ends, or we can say axonal ends, in the cytoplasm of the axon. Why the calcium ions have entered? Because that nerve impulse have lead to opening of these ion channels. There is change in the voltage gated calcium ion channels, they open, right? What are these? These are voltage gated calcium ion channels. So voltage gated calcium ion channels have been opened. Calcium fuses with the synaptic vesicle. What is this? This is synaptic vesicle. And when the synaptic vesicle fuses with the membrane, it will release the chemical. You call it as neurotransmitter. Neurotransmitter. So the neurotransmitter have been released in this space, and the name of the space is synaptic cleft. What do you call it as? Synaptic cleft. And the entire junction is synapse. It's a chemical synapse. The entire structure is chemical synapse. The space between the two neurons is synaptic cleft. And the neurotransmitter is released in the synaptic cleft. Now, for example, we want to excite this neuron. So accordingly, a neurotransmitter will be released. For example, if I say the acetylcholine is released, and this acetylcholine, or we can say adrenaline, better say adrenaline. Adrenaline is a hormone and it is also a neurotransmitter. So here the adrenaline is released, it will bind to these channels, and these channels will lead to the entry of sodium ions. And whenever there is entry of sodium ion, that causes depolarization, and whenever there is depolarization, there is excitation. There is excitation, right? And now imagine a neurotransmitter is released, that is dopamine. What is it? Imagine that's dopamine. Dopamine is inhibitory neurotransmitter. Now if this one attaches, it will always attaches to potassium ion channel, and that will lead to the exit of potassium ions. And when potassium ions leave, that state is repolarization. And when there is repolarization, there is always inhibition. That is not excitation. During repolarization, it is going to be into rest state. That means it will not do anything, it will be inhibited. And in the depolarization, it will be in the excitation state. So here we learned that with the help of a chemical synapse, we can generate two types of responses. One is inhibition, one is excitation. For example, if I want to contract my muscle, I will be releasing an excitatory neurotransmitter. Imagine it is adrenaline, and that will lead to opening of sodium ion channel, and my muscle will get the nerve impulse and it will contract. Now, if I want to stop the muscle from contracting, my brain is sending dopamine or any inhibitory neurotransmitter, and my muscle relax, right? So this is how different types of neurotransmitter, they can lead to the different type of responses. That's a positive point of chemical synapse. What's about the electrical synapse? In the electrical synapse, there are two neurons and both are attached to each other, and in between them are present gap junctions. What are present? Gap junction. These gap junction can lead to bidirectional flow of ions, right? So from here, sodium ions or potassium ions, they can easily move in or outside. That can, imagine if these both are sodium ions, these both can be moved. And as a result, the wave of depolarization can move from one neuron to another, right? So what do you think? Will they be faster or this will be faster? If I say in the electrical synapse, there are just junctions present. What are these junctions? Gap junction. So if I say the stimulus have arrived on this neuron, if this one receives the stimulus, this can easily cause the depolarization of this one by sending the sodium ions. So simply, there is nothing to do. You don't have to fuse calcium ion with the synaptic vesicle, no release of neurotransmitter. They're just simple. And hence, they will be little faster. They will be little faster than chemical. But chemical one have a very important role, and that's a benefit. They can generate two type of responses according to the use of the neurotransmitter. So it's kind of a better one. Fine. But the faster one, they are the electrical synapses. So that's all about the synapses, guys. Let's move further. Let's move further and solve this question. The postsynaptic potential is always excitatory, always inhibitory, both excitatory and inhibitory, none of the above. So postsynaptic one can be both excitatory and inhibitory. We have done right now. So this potential that have arrived here in the postsynaptic neuron is also known as postsynaptic potential. Okay, so this postsynaptic potential, they can be excitatory as well as inhibitory. Fine. Next. Okay, let me write the answer. Answer is 3. Next, guys. When a neuron is not conducting any impulse, that is resting, the axonal membrane is comparatively more permeable too. For example, this is minus 70 millivolt. So this membrane is more permeable to more exit of potassium ions. So simple answer is 2. Next. Sodium potassium pump transport 3 sodium outwards and 2 potassium inward. 3 and 2, answer 2. Okay. So these type of questions are brainstorming question. That will make sure how much your concepts are, have been built nicely or not. The electrical potential difference across a resting plasma membrane is called the, very simple, what do you call it as? Electric potential difference across the resting plasma is resting membrane potential or resting potential. Next. Depolarization occurs due to influx of potassium ions, efflux of sodium ions, influx of sodium ion. Very simple. Whenever there is stimulus, it leads to opening of sodium ion channels. There is influx of sodium ions. Sodium ions enter inside. So influx of sodium ions causes depolarization, and efflux of potassium ions causes repolarization. Okay, all right, next. So next we are going to talk about your brain, and let's get started. So let's get started with brain. So if I talk about brain, this is a very important part of your body, and it's a part of central nervous system. And this complex structure has been divided into 3 parts. How many parts, guys? 3 parts. So the brain has been divided into 3 parts. First is forebrain. Second is midbrain. And the third one is hindbrain. Right? So it has been divided into 3 parts: forebrain, midbrain and hindbrain. In forebrain, we have structures which are cerebrum, diencephalon, and olfactory lobes. Right now, I'm just telling you the names of the structure which are under different parts of the brain. We'll also study the detailed diagram, so do not worry, okay? Then we have in midbrain cerebral peduncles and corpora quadrigemina. Corpora quadrigemina, because its name is like quad. Quad means 4. So it consists of 2 superior colliculi and 2 inferior colliculi, right? So how many total structures are there? In corpora quadrigemina, 4. That's why its name is quadrigemina. Quad means 4, right? So it consists of, or it is made up of 2 superior colliculi and 2 inferior colliculi. Then we have in hindbrain pons, cerebellum and medulla. Cerebellum, pons and medulla. Right? Midbrain plus hindbrain except cerebellum is known as brain stem. What do you call it as? Brain stem. Right? Midbrain plus hindbrain except cerebellum. The cerebellum does not count in this. This is known as brain stem. Let's see the structure of brain first. So as I've told you, we have 3 parts in the forebrain. One is your cerebrum. So this entire structure, highly folded structure, and the largest part of the brain is known as cerebrum. And then we have diencephalon, and diencephalon is made up of three portions. One is thalamus, other is hypothalamus, and then this portion, this one here, this is epithalamus. This is epithalamus. So these three structure, they collectively known as, these three structure, they are collectively known as diencephalon. What do they known as? Diencephalon, right? So if you can see, this portion is epithalamus, center is thalamus, and down this portion is hypothalamus. So they are present in a manner that means above the thalamus, if this is a thalamus structure, above the thalamus is epithalamus and below the thalamus is the hypothalamus, fine. And then we have this portion, this one, which is the midbrain. And as I've told you, midbrain is made up of corpora quadrigemina and cerebral peduncles. This portion is the cerebral peduncles, and this portion, these one, they are the corpora quadrigemina, right? Because this is a section of brain, so you can see only two colliculi here. And then this is the center portion, that is cerebral aqueduct. We'll be talking about this in detail when we'll be discussing the midbrain. I'm just telling you where the parts are present. And then we have the hindbrain. Hind because it is present like at the last. Whenever we are talking about fore, that means comes first. Whenever I'm talking about mid, that's in the center. And if I'm talking about hind, that means it's at the last. So hindbrain consists of pons, and this portion is pons, this portion is medulla, this one entire portion is cerebrum, right? So as you can see, or if you have noticed here, this midbrain, pons and medulla, they look like a stem together. They look like a stem of a plant. So that's why they are known as brain stem. They are present togetherly one after the another in a manner, in a, you can say in a linear manner. So you call it as a brain stem. And then medulla oblongata, also known as oblongata, right? You must have done this in the junior classes. It will elongate to form spinal cord. It will elongate to form spinal cord. Okay. So these structures, you must have heard of it. Pons is also known as pons varolii, right? And medulla is also known as medulla oblongata. Fine guys, so do not worry about that. Sometimes you use just word medulla and sometimes you use the word medulla oblongata complete, right? Okay, so this is the positions where they are present. Now let's talk about how your brain is present. Or for example, most of you must have a dream to become a neurosurgeon, right? Are there any people who wants to be a neurosurgeon? Then put a thumbs up in the comments, right? So, for example, you're doing a surgery of a brain, then what will you do? Or you must have seen in the movies. Well, first of all, whenever a person undergoes brain surgery, that person's, head is shaved. So, first of all, there is a layer of hair, and then there will be a layer of skin. After skin, you have to go inside that structure, right? You have to reach towards brain, and then you have to cut the skin, and below the skin is present your skull or the cranium, right? This is how it is. And then when you open the skull, you know how do they open the skull? With the help of a heavy machinery, which is used to cut the, these, woods. So that machine is used to cut the brain skull because the skull is very strong. Nature has given you very strong skull. So now they will cut the skull and they will move inside. Now what's present inside the skull? So straight away the brain is not present inside the skull. Rather, the brain is covered by certain protective coverings. You heard it right. Brain is covered by certain protective coverings. You call them as cranial meninges. Meninges are protective coverings. Cranium because they are the part of cranium, right? Protective coverings. Okay, let me show you. First of all, for example, you will have this layer of skin here. Then after skin, what will be present? The skull or the bone portion. Then after bone will be present your cranial meninges. So there is first of all the outermost covering, which is very durable. So we call it as dura mater. Then comes the layer which is very thin and web-like. Web-like, the web of a spider. And then comes another layer which is highly vascular, and this dips deep inside the brain. This dips means to enter. It will enter inside the brain tissue. And imagine these are your brain tissues here. Okay, so this is your brain tissue. This is your brain tissue. So this is how we have three coverings, three cranial meninges, guys. The first one, this one, is dura mater. This is the strongest layer, as its name suggests, durable. This will be durable. This will be very strong. And then comes the layer arachnoid mater, which is web-like. It is very thin and avascular. And then comes the pia mater. Pia mater is the innermost layer, and this enters brain tissues. Okay, so might be they will ask you a question about the sequence of these cranial meninges. So what these three layers are? These three, they are cranial meninges. So if they're asking you about this sequence from outside to inside, then what will you say? You will say first dura mater, then arachnoid mater, then pia mater, right? Now there are certain spaces present between these two. So these are the spaces that are present between dura mater and arachnoid mater and pia mater. Now what are these spaces? So this space, this one, is known as subdural space. And this space is known as subarachnoid space. Fine. Now these were the three very powerful coverings that protects your brain. Okay, guys. All right. Let's move further. And before starting the forebrain, okay, let's read the NCERT. The brain, okay, let me just increase the font size. The brain is the central information processing organ of our body and acts as the command and control center. So who controls you? Not your heart, your brain. So that's the brain that leads you to do sometimes funny situation, sometimes hard situation, put you in the hard situation, and sometimes into very embarrassing situations as well. Okay. It controls the voluntary movement, balance of the body, functioning of vital voluntary organs, thermoregulation, hunger and thirst, circadian rhythms of body, activities of several endocrine glands and human behavior. So all these functions are done by your brain, and we'll be studying by the types of the parts of the brain, what part of the brain performs which function. Okay. Then it is also the site for processing of vision, hearing, speech, memory, intelligence, emotions, and thoughts. The human brain is well protected by the skull. Inside the skull, the brain is covered by cranial meninges, consisting of an outer layer called dura mater, a very thin middle layer called arachnoid mater, and inner layer which is in contact with the brain tissue is pia mater. The brain can be divided into three major parts: forebrain, midbrain, and hindbrain. Fine. So that's about your introduction of the brain. Let's talk about the very important part, the first part, that is cerebrum. So we are going to talk about this highly complex portion of your brain, the largest part of your brain. Okay. So you will be also shortly talking about these portions. You might be wondering, ma'am, you have not told us about these two portions. I have left these portion for the cerebrum. Okay, so don't worry, I know everything. Okay, so let's get started with the cerebrum. As I have said, the cerebrum is the largest part, the biggest portion, highly folded. There are a lot of folds, right? As you can see in this diagram, there are a lot of folds here. These are folds. Why there are folds? You'll get to know shortly. So it's the most complex and it is seat of intelligence. If we'll cut the section of the brain, you will see two regions, one outer region, another inner region. The outer region will be of gray color, whereas inner will be of white color. So you call that as gray matter and white matter. Okay, for example, if you have cut the brain, you will see on the outside there are certain gray color matter present like this in the form of 6 layer of neurons. And inside you will find there is presence of white matter like this. Okay, so this portion is gray matter, and gray matter is present in the form of 6 rings or layers of neuron, whereas white matter is the one which is white in color. Now, why white is white and gray are gray? The reason is, For example, you have collection of neurons in the brain because brain is the part of nervous system and this is made up of neurons and this is made up of neurons. Now imagine this is your neuron. This is how neurons are, right? The gray matter contains all the cyton and the group of cyton in the central nervous system is known as nuclei. What do you call it as? Nuclei. So the group of all the cyton, group of cyton is known as nuclei. And this nuclei gives gray color. Why? Because in cyton we have Nissel's body and this Nissel's body provides gray color. What color? Gray color, because it has granules, it has ribosomes and that gives gray color. Okay, now this portion where you have axon, the collection of axons in the brain or in the central nervous system is known as tract. What do you call it as? Tract, right? So here if I say the group of axons is known as tract and this tract gives white color. Why? Because axon have myelin sheath and myelin sheath give white color. Myelin sheath give white color. That's why the color of gray matter is gray and the color of white matter is white, because it has a group of axon and this have a group of the cytons, and group of cyton in central nervous system is known as nuclei and the group of axons in the central nervous system is known as tract. Okay, all right. Now it is highly folded, as I've told you. Now what's the reason? If I say something is highly folded, that is always will increase the surface area. And if it will be good, it will be very good if you have more surface area. More the surface area, that will be more neurons will be there. More neurons, more intelligence, right? Okay. So it doesn't always means more neurons, more intelligence. Intelligence is also directly proportional to the how number of synapses are formed between the neurons. Okay. So whenever you learn something, a new synapse is formed in between your neurons. So this is like I am teaching you something. So when it will go to the permanent part of your memory, there a synapse will be formed. Okay, so whenever due to certain neurodegenerative disorders like Alzheimer's, these neurons get damaged, the synapses got damaged, and you will start to forget things. Maybe your memories, happiest memories, your sad memories, or your memories of your childhood, you will not be able to remember that. Okay, all right. Anyways, so as you can see, your brain is divided into two hemispheres. How many hemispheres? Two. So there are two hemispheres of the cerebrum, not the brain. I think I said brain earlier. One is the left one, another is the right one. Left one and right one. Okay, let me show you. Let me show you here. Okay, please open this up for me. Okay, so as you can see in this diagram, guys, this is a brain, this is cerebrum. Do not just look at the different colors. This is one hemisphere of the brain, this one. This is, imagine, the left hemisphere, and this one is the right hemisphere, which is not drawn here. Okay, so this is the only one hemisphere that have been drawn, the entire one. This is left hemisphere. Both the hemisphere are somehow connected with something. This is blue color thing. So this blue color is nothing but these are basically group of neurons which are connecting both the hemispheres together. For example, this is how your brain is. This is the left hemisphere, this is the right hemisphere, and both are connected to each other with some nerve fibers, and you call them as, and this entire structure is known as corpus callosum. What do you call it as? Corpus callosum. Okay, so here if you have seen in the NCERT diagram, I have shown you here, this is one hemisphere. This is one cerebral hemisphere, and this is corpus callosum, which is connecting both the cerebral hemispheres together. Okay, so let's write this down. Two hemispheres are present, the left cerebral hemisphere and the right cerebral hemisphere. Okay, both are connected by corpus callosum. What is corpus callosum? It's nothing. These are nerve fibers. What are these? Nerve fibers. Okay. All right. Now, your brain has three areas. How many areas? Three areas. One is sensory, another is motor, and the third one is association. Based upon what type of a neuron or nerve fibers it have, whether it's afferent or efferent. In simple way, I'll say whether the portion of the brain is having sensory neurons or the motor neurons or the, both the neurons or the interneurons, depending upon that, your brain has different kinds of area, or we can say depending upon what type of impulse enters to that portion, you call or you have divided these into three areas. For example, if I say this is the left hemisphere, in the left hemisphere, this portion, it receives impulse for vision. Right now you are looking at me, that's the sensory impulse, because the eyes are sense organs. Okay, so sensory information is going to this particular area of your left hemisphere, so this portion is your sensory area. If I say this is a portion that will send the impulse to your skeletal muscle, so this area is leading to the motor activities, so this will be your motor area. And then there can be certain areas known as association areas. These areas are the one, like this is sensory area, this is motor area, and this is association. The function of association area is to associate these signals. That means it will read the sensory information and then it will convert it into motor impulse. For example, the sensory information is like someone called you, so you hear something, this is sensory information, went to sensory area, and now you have to move towards that person. So now this information may go to association area. Association area will read the sensory information, it will read the sensory information and convert it into motor, and later on it will convert it into your memory, and it can also make it the part of your communication. Right? So this is one what association area does. So sensory areas, it will receive the sensory input, any sensory information, vision, taste, or other sensory like your hearing, all the sensory information like touch, cold, temperature, all these sensory information will go to, it will go to your sensory area. Motor is related with the motor activities, right? Whereas association, it will read the sensory information, convert it into motor, and also it will make it the part of memory and communication. Okay, just for example, I always give this example, it's a kind of funny one also. For example, there are two friends and somehow they are fighting for something. Okay, for example, maybe a candy. Okay, so one friend slapped the other one. For example, I'm the other friend and I had a slap on my face. So this is the sensory information. This now went to my brain. Now in my brain, the motor one will read it. Okay, motor one will read it and convert it into motor, and I also slap the person. Okay, I also slap the person. So also now this will be stored in the memory form in my body, in my brain. And it will then can also become the part of my communication, and I will tell everyone that that my friend hit me, I also hit that person. Okay, or it will also be a part of my memory, and I will not talk to that person tomorrow. Why? Because we had a fight, and my association area has converted that into memory form. Fine, but you never fight with anyone, right? Okay, so that human is the only person you call it as a true human who has control over their senses. Any person who easily got angry or easily got a lot of emotional over things, or easily get into some fighting, and so these are people who do not have control over their senses. If you are a very kind person, you are fine with everything, and you are very calm, you think and accordingly do your things in the situations accordingly, then you are a very calm person. That means you have control over senses. So it's very easy to become a person who can fight easily, who can get angry easily, but it's very difficult to become the other one, the calmer one. A calmer person is the one who has control over the senses, and this is a very important and difficult task to do for a human being, right? So this is how the monks are. The monks, you will always see, they are very calm, right, whatsoever the situation is. So this is a reason, because they have the control over their senses. All right, anyways, a lot about this. Let's move further and talk about what are the various four lobes of the brain. There are total four lobes, right, or our brain has been divided into four lobes. So your brain is divided into four lobes. How many lobes? Four lobes. So one is frontal, second is parietal, temporal, and occipital. So the name is given according to the bone they are present beneath, right? For example, if I say this is the diagram, okay, that's a 3D structure. This green one is your frontal lobe. This one is your parietal lobe. This one is your temporal lobe, and this one is your occipital lobe, right? So we have divided them into the lobes, and they are performing various different functions. So the areas can be different. For example, if I say the temporal is performing the function of the interpretation of sound, that means every sound impulse will go into your temporal region. So that's a kind of a sensory area it contains, right? And this one will also contain the sensory area, the occipital lobe, because it is also associated with the sight. Again, the work of sense organ. Fine. Then we have parietal, which is related with your sensation of touch, pain, and cold or hot. So, for example, if you are touching something hot, this sensation will go inside your which part of cerebrum? The parietal, or the memory of anything will be stored in this portion. For example, if I say these things are hot, anything is present on the stove will be hot, so that means somewhere parietal lobe have that memory. Okay, and then we have the frontal one. In frontal one, it controls all your logic, reasoning, all your voluntary activities. For example, I'm moving my hand. I'm moving my hand. So this impulse have been sent to my skeletal muscle through this portion of my brain, that is the frontal lobe. Okay, that is the frontal lobe. So any logic, reasoning, creative ideas, your intelligence, IQ, EQ, everything is controlled by this portion of your brain, and that is frontal. All right, guys. Okay, moving further to the next portion, that is diencephalon. So diencephalon is basically, it is divided into three portions. The one which is present at the center is thalamus. Above the thalamus is present epithalamus. And then we have below here the hypothalamus. Right? So here first of all we talk about epithalamus, this one. First one, epithalamus, thalamus and hypothalamus. Okay. So this one is forming the roof of your diencephalon, and it also contains a gland which is pineal gland. So the name of this gland is pineal gland. This pineal gland, it secretes the hormone melatonin. What hormone it secretes? Melatonin hormone. And this one controls your sleep and wake cycle. Also call it as, you call it as circadian rhythm. What do you call it as? Circadian rhythm. What is a circadian rhythm? Your body is functioning in the form of a cycle, like you sleep during night and wake up in the morning. So there is a body cycle is set for a lot of number of things, like in female a menstrual cycle is going on, right? So just like that, your body is in the form of cycle, and thanks to this hormone which is melatonin. Okay, then we'll study this gland in the chapter chemical control and integration. This is a highly vascular portion, that means it will contain a lot of blood vessel. Then we have the thalamus, which is responsible for touch, temperature, pain and pressure. All these impulses will first go to this thalamus portion. Then it will send to the cerebrum portion for storage for memory. Okay. Like your hot sensation will first go to thalamus, and then it will go to your cerebrum's portion, which is the parietal, and then it will be stored in the form of memory. Okay. Then it has maximum number of neurons. Then we have hypothalamus. It forms the floor of your diencephalon, and this is also highly vascular. So this is also a kind of endocrine gland. Why? Because it secretes hormones. So next thing that we are going to discuss is the functions of hypothalamus, which is very important. So the first function is, as it is an endocrine gland, it secretes hormones. So it secretes two types of hormones. Okay. So for that, first let me tell you, if this is hypothalamus, hypothalamus is connected to a gland known as pituitary gland. Which gland? Pituitary gland. Epithalamus contains pineal gland. Hypothalamus is attached to pituitary gland. Okay. Now, so the hormones secreted by the hypothalamus are of two types. The pituitary has two areas. This is anterior pituitary. This is anterior pituitary, and this one is posterior pituitary. Okay. So what does hypothalamus do? It will secrete certain hormones like oxytocin and ADH, which will then travels and they will be stored in the posterior pituitary. So first kind of hormones that hypothalamus secrete is the one which will get stored in the posterior pituitary. Fine. Second type of hormones it will secrete that will control the functions, that will control the functions of anterior pituitary, right? So here, for example, the hormone is a growth hormone which is secreted by anterior pituitary. So hypothalamus may release an inhibiting hormone or the releasing hormone. If hypothalamus is secreting the releasing hormone, the growth hormone will be secreted by the anterior pituitary. But if hypothalamus is secreting inhibiting hormone, the growth hormone will not be secreted by the anterior pituitary. So in a way, it is controlling the pituitary gland. So it is producing two type of hormones. One is ADH and oxytocin. ADH, another name is vasopressin. We have discussed this in excretion. So ADH and oxytocin, these one, they are stored in posterior pituitary. All right. Whereas it also secretes inhibiting and releasing hormone that controls anterior pituitary. That controls anterior pituitary. Okay. All right. So what are the other functions performed by hypothalamus? Second, it acts as a thermostat. So thermostat is something that will help in thermoregulation. That means control of temperature. So it also controls your body temperature. Whenever you have certain viruses or any bacteria or any infection in the body, your body temperature increases. So why it is increased, why the body temperature has been increased? Because during immune action, certain substances are released that will go to hypothalamus and ask hypothalamus to please increase the body temperature. So if the body temperature is increased there, hypothalamus will got it to know that the body temperature has been increased and it will try to regulate it also. That means sometimes it will also lower this down. So this is how it can increase or either decrease the body temperature. Third thing is it has a center for hunger, thirst and satiety. Satiety means satisfaction. For example, you have had your food and your tummy is entirely full. You feel satisfied. Very nice. Or if you have had something which is your favorite, like some people like, dosa, some like rasam rice, some like rajma chawal, or, anything pizza or something. How do you feel after eating it into a full state of your stomach? You feel it very nice. So that's a kind of satisfaction you have. So that center is also in your hypothalamus. Fourth, it also controls your body cycle, which is circadian rhythm. Circadian rhythm, now you must be wondering, ma'am, it is controlled by the melatonin hormone. Very true. Melatonin hormone is the one which will be secreted, but what will be the center, control center? That will be the hypothalamus. Also, it controls your sexual and emotional behavior. You are angry, you are sad, you are happy, you are in love. It's all due to your hypothalamus, not just because of your heart. You never fell in love according to heart. You fell in love due to your brain. Yes, according to brain. Okay. All right. So this is what your diencephalon is. Let's talk about a very important aspect because we are talking about emotional brain, and that is your limbic system. So limbic system is the emotional brain. That's the emotional brain. That's the emotional brain. That means it will control all your emotions. So deep inside the cerebrum, there are present a number of small nuclei, and sometimes you consider them as a separate part. For example, there is a fork-shaped structure formed inside the cerebrum. Okay, so inside the cerebrum, deep inside the cerebrum, these structures are present. For example, if this is a corpus callosum, then here you can see a septum nuclei. What is a nuclei? Group of cyton. Okay, and there is one portion amygdala. This is like your hypothalamus, and down here is present hippocampus. All these structures that together form what do you call it, as a system in the body known as limbic system. Okay, now apart from that, there are also present just right here olfactory lobes. What are present? Olfactory lobes here, olfactory lobes. And in the hypothalamus are present small bodies which look like mammary glands of the human, and you call it as mammillary bodies. What do you call it as? Mammillary bodies. Though it's a part of hypothalamus, so sometimes it can be written separately also in the question. Are mammillary bodies the part of limbic system? Then you will say yes, mammillary bodies are definitely the part of hypothalamus. These are one paired, looks like a breast of human, and their function is in olfaction. What is its function? Olfaction. So all this is controlling your emotion and sexual behavior. So this entire structure is known as limbic system. Okay, for example, you must have watched on television a lot of advertisement of the perfumes, right? I will not name any perfume, but yes, and they show that when a male sprays a perfume and a female got attracted. This is absolutely at some point true. Yes, a female is attracted by the olfaction, but it doesn't mean the female is attracted to the man. Right, the female is attracted to the olfaction, but not to the man. For example, if the olfaction is good and they go there and see, oh, she doesn't like a man, doesn't mean if she likes olfaction she'll like the man, right? So yeah, olfaction is one kind of a thing that will connect two couples together. Also, there is a kind of olfaction of every person have their own different kind of order of a body. So yes, why that is important? Because it then goes to the mammillary glands and olfactory lobes. Olfactory lobes will collect the information of olfaction, send it to the mammillary bodies, and then it will send the information to the limbic system. And then this is how the emotional and sexual behavior will be enhanced, right? So it can enhance that property, but it doesn't mean that's the cause of that, right? Okay, so the function of septum nuclei, this will always be active during sexual behavior. All right, amygdala is the one that is responsible for anger. So any people who have anger issues, it has been the study was done, and it has been stated that anyone who has anger issues, their amygdala secretes high amount of neurotransmitter, and amygdala can be the larger in size. Usually in the criminals who have done so heinous crimes, in those people the amygdala is in the larger portion or the larger size with a lot of neurotransmitter activity, right? Then we have hippocampus. The function of hippocampus is to convert short-term memory into long-term. Short-term memory into long-term. For example, right now, you are studying all of these things from me. Everything is in the form of short-term memory. But later on, it will be converted into a long-term memory when it will pass through hippocampus. So guys, you have to revise again to stimulate the hippocampus so that the things should go in the long-term memory, right? Okay, that's about the limbic system. Let's talk about the midbrain. So this midbrain, this is how it looks like. As I've told you, it consists of two portions. One is cerebral peduncles, another the corpora quadrigemina. So two superior colliculi, two inferior colliculi, they collectively form corpora quadrigemina. Now what are the functions of these organs? First of all, corpora quadrigemina is present on the dorsal side, whereas the cerebral peduncles are present on the ventral side. The function of cerebral peduncles is, it's a group of neuronal fibers, and these nerve fibers or neuronal fibers, they are attached to the upper part of the brain, that's the forebrain. For example, this is forebrain, here it is a forebrain, and here is present the hindbrain. So in a way, we can say the midbrain is connecting forebrain with the hindbrain, because cerebral peduncles have a lot of neuronal fibers. Okay, it has a lot of nerve fibers. So cerebral peduncles connect forebrain and hindbrain. Okay, now What about these colliculi? This one is responsible for vision reflexes and the inferior one is responsible for auditory reflexes. Now what are these? For example, you're just sitting here or you're sitting outside your house, not on the road, near the gate. Never sit on the road. You may meet with some accidents. Don't do that. Nothing. You will never do that. Okay. So just you're standing or sitting outside your house. Okay, just near the gate. All right, away from the road. So imagine suddenly at a high speed a car moves like that. Now what will be your response? This will be your response. Okay, so this is the kind of vision reflex. Were you prepared for that? No, you were not prepared. So it was an uncontrolled thing. It was an uncontrolled thing towards the stimulus. And what was the stimulus? The thing, a vision, right? And because it's uncontrolled, that's a reflex. That means it was unplanned. You didn't plan for it. You never knew that a wonderful car at high speed will pass through that. So that's a kind of a vision reflex. Now suddenly you're standing outside your house and suddenly you hear that in the kitchen or somewhere in your house, a very large vessel or utensil fall down. You will hear, and what will your response? Like this. Okay, so this is auditory reflex. Why are you not moving outside your house like that? Because that's a kind of a reflex, and you just made that reflex just because of hearing, right? So that's a kind of auditory reflex. So these two type of reflexes, they're controlled by your midbrain. So if there is a statement in your exam that the reflexes are controlled by your midbrain, will you write yes or no? You will write yes. Reflexes are also controlled by your midbrain. What kind of reflexes? The vision reflexes and the auditory reflexes. Another question that they can ask is, what is the position of corpora quadrigemina or the colliculi? That's dorsal. Now we say they are 2 in number, these are also 2 in number. So see, we have done the dissection. So if one ball is present here, that is one superior colliculi, the another ball will be present right here. So 1, 2, 3 and 4 balls are there. So that's why they're corpora quadrigemina. Now inside them is present a small duct, and that duct is cerebral aqueduct or iter, which is the duct of Sylvius. Okay, what's the function of this duct? We'll be talking about that very shortly. All right. Okay, next we have our pons and medulla. Pons is the one which has a pneumotaxic center, and pneumotaxic center is the switch off center for breathing. All right. Also, pons is the structure that will connect your upper part of the brain, that is your midbrain, and lower part of the brain, that is your medulla, with the cerebellum. So how it is positioned? For example, if this is pons, this is medulla, right? This is cerebellum, and this is midbrain. All right. So this pons is connecting to the midbrain. It is also connecting to the cerebellum, and this is also connecting to the medulla. If this is pons, this is medulla, and this is cerebellum. So this is how the pons means bridge. Its literal meaning is bridge. So it's a kind of a bridge between all these structure. So it connects to midbrain and hindbrain. All right. Now, talking about the medulla. Medulla have a lot of centers. It has a centers, we'll start from the digestion chapter. It has a center for vomiting, guys. It has a center for cardiovascular system. How can we forget if there is pneumotaxic center? Here we have respiratory rhythm center. All right, we also have hiccuping center here. Hiccuping center. Right? So all these centers are present in the medulla, and medulla elongates and then it will form spinal cord. Medulla elongates to form spinal cord. All right. Okay, so the last part in the hindbrain that we have is the cerebellum. So cerebellum is known as little cerebrum. Why? Cerebrum is the largest part of the brain. This one is the second largest part, right? Another thing to note down here, cerebellum, just like that of cerebellum, just like that of cerebrum, is also folded. It's also folded. So it's the second largest part, and it is also folded. What's the function? Its function is in equilibrium and balance of the body. The function is equilibrium and balance of the body, right? So if you'll see the cerebellum structure, let me show you the diagram here, guys. This one, you can see this structure. Here outside is present the gray matter, inside is present the white matter. So this arrangement of gray matter in white matter is known as arbor vitae. Let me draw the diagram clearly by the hands, obviously, mine hands. So here, if you'll see, this is a gray matter. Okay, this is a gray matter, entire gray matter. The white matter is not present like that it was present in cerebrum. In cerebrum, what was the scene? Outside there was a gray matter, inside there was a white matter. Okay, this was in the case of cerebrum. But in case of cerebellum, the gray matter is dispersed like that, and in between the gray matter is present white matter. And it looks like the branching of a tree. You call it as arbor vitae. So arbor vitae is the tree of life. What is this? You call this as arrangement of white matter in the gray matter. Arrangement of white matter into gray matter. That's what arbor vitae is. And what's the function of your cerebellum? Equilibrium and balance. For example, I'm standing erect like that, right? Just straight. I'm standing straight. I'm not falling here and there. All thanks to my cerebellum. It controls all these balancing and equilibrium. And it also controls all your voluntary activities. How? Voluntary activities are initiated by your cerebrum, but then they're coordinated by cerebellum. So it also coordinates voluntary actions. It also coordinates voluntary actions. For example, like I started dancing. This is what my cerebellum has started and giving the information. Okay. So now what I'm also doing this and that. Okay, just like that or something like that. So my coordination is between my steps. That's it's done by or it's controlled by my cerebellum. But the initiation of voluntary activities, it's done by the my brain cerebellum. But its coordination is done by my cerebellum. Okay, that's about the cerebellum, guys. Now let's talk about ventricles. What are ventricles? Now here we are going to talk about two things. One are ventricles, another is CSF. Okay, ventricles are the spaces present in brain. Yes, there are spaces present in brain that contains CSF. That contains CSF. Now, what is CSF? It's the cerebrospinal fluid. So like your blood is your body fluid and it's present in your entire body organs. But brain and spinal cord have their separate fluid known as CSF. And the CSF is formed from blood only. Formed by filtration of blood. So when the blood got filtrated, it forms the CSF. Okay. Now, why there is a need of a separate fluid? Why can't the blood be the body fluid of your brain as well? The reason is the blood might contain a lot of harmful substances like it can be microbes, it can be antibodies. So these substances can actually damage your neurons. And you know, once the neurons are damaged, they cannot be healed. And neurons are very important. Imagine one neuron get damaged and it has formed synapses with another neurons. So a lot part of your memory got lost. So we cannot take that risk. So for nature has made another fluid for brain by filtration of blood only, that's CSF. So how is it formed? It is formed by filtration of blood through blood-brain barrier. If this is the blood and a blood vessel, then there is a blood-brain barrier. This is a blood-brain barrier. From here, the blood will be filtered and this will be now known as CSF. What will you call it as? CSF. And this place are the ventricles. So the blood when filters gets filtered out and the fluid CSF it's enter into these empty spaces known as ventricles. So for example, you have this cerebrum, left hemisphere, right hemisphere. So in the left and the right hemisphere, two empty spaces are present. You call them as left and right ventricles. What you call it them as? Left and right ventricles or the first ventricle. Okay. Then these are the ventricles of your cerebrum. So these one ventricle is present, or each cerebrum contains each ventricle. Okay. So they're present in cerebrum. So they contain the cerebrospinal fluid. Now comes to the another portion here. There comes the another, okay, let me drink some water because I'm very thirsty. All right. So these left and right ventricle, it will open into another ventricle, which is your third ventricle. Why this is third ventricle? Because first and second are these one. They are also known as lateral ventricles. They are also known as lateral ventricle. So the ventricles from the cerebrum, all these are connected. They are leading to the flow of CSF. Okay. It will open into third ventricle, which is around your thalamus and hypothalamus. And then it will open into iter or cerebral aqueduct, which is the part of your midbrain. And this is your midbrain. Okay. And this duct is your, yes, iter or aqueduct. Now this will now open into the another duct, that is your fourth ventricle. So this one is the fourth ventricle. This is around your hindbrain. This is around your hindbrain. Okay. And now this has certain openings here. It has three openings, 1, 2 and 3. Right. So the cerebrospinal fluid, so here we have cerebrospinal fluid. It will, from here, it will go to the spinal cord, because spinal cord is also hollow inside. It also have certain spaces through which the fluid can move, because spinal cord is also important. So you also cannot give your blood to it. And then from here it will go to subarachnoid space. Which space, guys? To subarachnoid space. Same for the another one, to subarachnoid space. Alright, so this is how the subarachnoid space, the one in the cranial meninges, it also have the CSF, cerebrospinal fluid. Okay, because from here it goes to the subarachnoid space. So this is how these empty spaces contains one fluid. The name of the fluid is cerebrospinal fluid, formed by the filtration of the blood. Alright guys, that's about your ventricles. Let's talk about spinal cord. So spinal cord is rod-like structure. It's a rod-like structure, but it's hollow and it contains CSF. Just like that of brain, it also have meninges, spinal meninges. They are just same as like that of brain, like dura mater, arachnoid mater and pia mater. They all will be present, right? So if we'll cut this section of the spinal cord, you will see inside this hollow cavity will be present, which contains CSF. And the name of this cavity is central canal, right? And now you will see one opposite thing, opposite to the brain. In the brain, the outer area was of a gray matter and inner was of a white matter. Here there is opposite. This area is a gray matter. This is a gray matter, gray matter, gray matter. Okay, and this one is a white matter. Outer area is a white matter. A very important thing that you need to remember here, that this spinal cord have two areas and both perform different function. Like this area is the dorsal side. Dorsal, posterior, or it can also be known as sensory side. Okay, and then here this is ventral, motor or anterior side. Okay, you can learn it through DPS. DPS is Delhi Public School. These schools are very famous in Delhi, right? So Delhi Public School, dorsal posterior sensory. So dorsal side of a spinal cord is always sensory and ventral side is always motor. Once the question was asked in the NEET, so you must know that. Why it's sensory? Because from the dorsal side emerges the sensory neurons and from the ventral side emerges the motor neurons, right? And on the dorsal side is present here a root ganglion. What is present? Ganglion, and that contains pseudo-unipolar neurons. Now, these both cluster of neurons will join, and they will form a common nerve. And you call it as spinal nerve. What do you call it as? Spinal nerve. So we say that spinal nerves are mixed in nature. What is mixed in nature? That means it contains the neuron of both the sensory ones, or it contains both type of neurons, the sensory neurons and the motor neurons. Okay, and this is known as ganglion. You also sometimes call it as dorsal root ganglion. So that's the spinal cord, and from every side the spinal or from both the sides the nerves will arise, the spinal nerves. So spinal nerves, we say they are in pairs. How many spinal nerves do we have, guys? We have 31 pairs of spinal nerves. 31 pairs of spinal nerves. Fine. So that's about your spinal cord. Why I am teaching spinal cord in detail? The reason is once question was asked in the NEET, which side is sensory, which is the motor. And also this will be very important for you to understand the reflex action as well. Right? Okay. Anyways, move further and solve these questions with me, guys. An inner layer which is in contact with the brain tissue is called, very simple, not the dura mater, it's the outermost durable. Arachnoid is very thin. Pia mater is the one which innervates the tissues. So answer is 3. Next, the cerebral hemispheres are connected by a tract of nerve fibers called, very simple, corpus callosum. Corpora lata is the gland in the arthropods. Corpus spongiosum is present in the penis. This is also present in the penis. Okay, that's the connective tissue of penis. Answer is 1. Next, which of the following is a function of hypothalamus? Act as thermostat. True, it controls body temperature. Also thirst center, and it also controls emotions. So all of these, all are the functions of hypothalamus. Next, okay, that's the repeat, I guess. Midbrain consists mainly of 4 round swellings, lobes called corpora quadrigemina, that are present ventrally, dorsally, anteriorly, both 1 and 2. So straightforward, they are present dorsally. Answer 2. Okay. All right. Next. Which of the following center is present in medulla? Pneumotaxic, thirst, hunger, vomiting. You know that. It's in the pons. Thirst and hunger, they are in the hypothalamus. And this one is in the medulla. So answer is 2. Okay. Next. So next we're going to talk about is autonomic nervous system. So let's get started. So autonomic nervous system is also known as visceral nervous system. This nervous system, it innervates visceral organs. That's why you call it as visceral nervous system. It is of 2 kinds. One is parasympathetic and another is sympathetic. Parasympathetic and other is sympathetic. And both work opposite to each other. Now, how they are arranged in our body and where are they present? Because virtually you just always hear there are these type of system. This is peripheral, this is CNS, but you are not able to picturize thing until or unless you have an image of it, right? So let's see how they are present in the body. So for example, this is your central nervous system. Okay, and from central nervous system, I'm talking about brain and spinal cord. Okay, and I said that cardiovascular center is present in medulla, and that controls your heartbeat. It can either increase the heartbeat or it can decrease the heartbeat. Like I say, parasympathetic decreases the heart rate or heartbeats per minute, and this one increases the heart rate or heartbeats per minute. So how it is going, right? How it is doing. So from brain's medulla, the neurons of, for example, the autonomic nervous system will arise. It can be either a sympathetic or parasympathetic, depending upon what function they're performing, right? And now these neurons, they will form a synapse inside the ganglion. And these ganglions are present. It can be present near the central nervous system, or they can be present away from the central nervous system, right? And now the neurons, these neurons, what are these? These are neurons. The neurons or the nerves that are coming out from the central nervous system, for example, these are the neurons of parasympathetic nervous system. They will first form synapse with the ganglion, and then they will go and innervate your organ. Like this is the organ. Okay. For example, this organ is heart, and we want to decrease the heartbeat. So here the parasympathetic nervous system is functioning, and here the neurons of parasympathetic nervous system will first form synapse with ganglion, release a neurotransmitter. And what's the name of that neurotransmitter? Acetylcholine. Because you know the nerve impulse is transmitted from one neuron to the another with the help of release of a neurotransmitter that we have done already in the transmission of nerve impulse, right? Now when it has to go to the heart and it has to send impulse to the heart, what it will do? It will again release a neurotransmitter, that is ACH, again acetylcholine, and accordingly the function will take place. ACH will inhibit the function and heart rate will decrease. Okay, so we say parasympathetic decreases heart rate. Now imagine if these are the neurons of sympathetic nervous system. So sympathetic nervous system's neuron will first of all release acetylcholine in the ganglion. Now after release of acetylcholine, the information has been sent to the neuron, or neuron have been excited. Now this neuron will now send the information to the heart by release of a neurotransmitter, that is adrenaline. And adrenaline will increase the heart rate, whereas ACH, if it is released, it decreases the heart rate. So we say both these are antagonistic. What are antagonistic? That works opposite to each other, but these both nervous system will innervate the same organ, right? Okay, another such example like dilation of pupils and constriction of pupil, right? Like if I say, if the sympathetic response goes to your eye, it will cause the dilation of the pupil. You understand the pupil, na? That black thing that is in your eyes. And if parasympathetic comes, it will cause a constriction. All right, okay. So that's about your autonomic nervous system. Let's talk about reflex action. Reflex action. Now, what are these? Very important topic. Yes. It's the unplanned. You have not planned it. Sudden response. What kind of a response? Involuntary. To a stimulus. To a stimulus. Like in the junior classes, you have heard of hot plate experiment. If you touch a hot plate, then suddenly you will take your hand away from it. You have not planned it. You didn't know that plate will be hot. So that's a reflex. Now what happened at that point? The skin, like if this is a very hot glass, okay, so it's not, but it's very cold. So if this is a very hot glass and my skin is touching it. So in my skin, some sensory receptors are present, which through sensory neurons are sending information to my brain or my central nervous system. It can be brain, it can be spinal cord. Okay, most of the reflexes, they are having a spinal cord as their center. Okay, so this impulse, it went to the spinal cord. Now spinal cord will send information through motor neuron, and I will keep the glass away. That's the muscle action that have been performed by my motor neurons. That's the reflex action. But here it was planned, so that's not the reflex. Reflex is sudden, very fast. Okay, so here what are the organs involved? here, first of all, the sensory receptors will be involved. Through sensory receptors, the information via sensory neurons. Which neurons, guys? Sensory neuron. What is another name of sensory neuron? Afferent neuron. Will go to central nervous system, CNS, and this can be brain or it can be spinal cord. Like I have told you about the vision reflexes, their center will be midbrain. If I'm talking about the reflex like we have done right now, that will be in the spinal cord. Okay, then via motor neuron, also known as efferent neuron, the information will be sent to your organs. It can be, yes, it can be your muscles or it can be your visceral organs. Now you must be wondering, ma'am, visceral organs, muscles? Okay, from muscles we understand the skeletal muscle. It can also be a smooth muscle. Visceral organs? Yes, for example, you have had some food and that food enters into your stomach. So stomach receives the food and that's a kind of a reflex that has been sent to the brain, and this will tell the stomach glands to secrete its enzyme. So that's also a kind of reflex action. So the structure that have been formed here is also known as the reflex arc. So what's a reflex arc? All these structures involving forming an arc-like structure, you call it as reflex arc. Okay, now let's let me explain you with the another example, that is knee-jerk reflex. Okay, so imagine this is your knee, this is your thigh, this is your leg, this is your foot. Okay, and this is your knee, and these are your muscles. And listen to the names of muscle clearly. This is your quadricep muscle. This is your hamstring muscle. This is your hamstring muscle. This is the quadricep. And this is your hamstring. So whenever you are sitting by folding your leg, just like you must be sitting in the chair and you have folded your leg in this manner, you will feel that there is a pressure on hamstring and hamstring is contracted. So whenever there is a folding of leg, the hamstring will contract, and whenever you have to straight the leg, like when the leg is in this manner, the hamstring is working, right? And if the leg is straightened at this time, the quadriceps must be working. Okay, for example, you're walking and suddenly you feel a pain here, you feel pain in the knee, and you stop and you straighten your leg. This is what is knee jerk reflex. Or let's do it on the other thing by experiment. When a doctor hit its hammer with the on the knee, right? This hammer is not the hammer that you use to, dig the nails in the wall. That's not the one. If you will use that, the knee will be broken and the person has to be hospitalized. So do not perform this at home. Okay. So this is a doctor's hammer. So when the doctor strike the hammer to the knee without telling the patient, at this point, there are sensory receptors present here. These sensory receptors on the tendons, they're present on tendons. What are tendons? That connects bone to the muscle. So these sensory receptors, what they will do, via sensory neuron, they will send this impulse to the dorsal part of your spinal cord. So this is the dorsal part, dorsal, posterior, sensory. So all the sensory information via afferent or sensory neuron, it will go to your dorsal part of the spinal cord. Now this will form synapse. It will form synapse with the motor neuron or efferent neuron and send the impulse to the quadricep muscle, and quadricep muscle will contract and leg will straighten up. Leg will straighten up. This is one reflex. This is one reflex known as knee jerk reflex. So what happened? When they strike the hammer, sensory receptors, they will send information to the spinal cord via afferent neuron. They will form synapse with this neuron, which is efferent neuron, which is a motor neuron. And the motor neuron will causes the contraction of the quadricep muscle. Quadricep muscles contract during knee jerk reflex, and this leg, like this, it will straighten up. This will straighten up. Okay, now there is one more reflex going on. So this reflex is knee-jerk reflex, and this reflex have only one synapse that is formed in the central nervous system. So you call knee-jerk reflex as monosynaptic reflex. What do you call it as? Monosynaptic reflex. Because here only one synapse is formed, so you call it as monosynaptic reflex. Apart from this, one more reflex is going on here, which is the inhibitory reflex, but you will not call it as knee-jerk reflex. Here, first the information is sent to the interneuron, and then interneuron will send the motor neuron to the hamstring, and it will causes the inhibition of hamstring. Because if the hamstring muscle will keep on contracting, the leg will always be folded. Okay, so there are two reflexes going on. First, this is knee jerk reflex, and this one is inhibitory reflex. In the inhibitory reflex, our only purpose is to inhibit the hamstring muscle, because hamstring muscle, if contracts, it will always lead to the folding of the leg, and if it will stop, the legs can easily be straightened up by contracting the quadricep muscle. Okay, now there you can see there are two synapses. So inhibitory reflex is a kind of polysynaptic reflex. If someone asks you what type of a reflex is a knee-jerk reflex, you will say it's a monosynaptic. Only one synapse is formed in the CNS. What is then polysynaptic? Why there are number of synapses formed? That's a kind of inhibitory reflex. One synapse is formed between the sensory and the interneuron. This neuron is the interneuron. And another synapse is formed between interneuron and the efferent or the motor neuron. Okay. All right. So let's read the NCRT, guys. Let's read the NCRT. Okay. So here you can see, you must have experienced a sudden withdrawal of a body part which comes in contact with objects that are extremely hot, cold, pointed, or animals that are scary or poisonous. So always you get a kind of a, move your body parts away. You move your body parts away whenever you kind of a fear or something. For example, if suddenly you put a hand on a bee, and that's a honeybee, you will put your hands away because you have a fear of sting. Or accidentally you are touching something, it's a rope, but then you find out it's a snake, then you will take it away. So it's also kind of reflex. Okay, so the entire process of response to the peripheral nervous stimulation that occurs involuntary, that is without conscious effort. That means involuntary or thought, and requires the involvement of a part of central nervous system, is called reflex action. So CNS and PNS both are working in reflexes. The reflex pathway comprises of at least one afferent neuron and one efferent. So as we have done, at least one afferent and one efferent should be involved here. Neurons arranged in a series. The afferent neuron receives signal from a sensory organ and transmit the impulse via dorsal nerve root into the CNS. So here we can see, this is what it say. The information sensory arises from sensory receptor, goes via afferent neuron and enter to the dorsal root of your sensory or the central nervous system. And here in the central nervous system we have spinal cord. Okay, now the afferent neuron receives signal from a sensory organ and transmit the impulse via dorsal nerve root into the CNS. The efferent neuron then carries signal from CNS to the effector. What is the effector organ? Your muscles, right? The efferent, that is motor neuron, will carry information from the CNS to your organ like effector organ like muscles. The stimulus and response thus forms a reflex arc as shown below in the knee-jerk reflex. You should carefully study figure this to understand the mechanism of knee-jerk reflex. So we have already carefully studied this image of a knee-jerk reflex. Fine. So that's your reflex action, guys. Let's talk about the sensory receptors now. So let's get started with the sensory receptor. Now, what are sensory receptors? These are small proteins which are innervated by neurons and they receive the input of sense, right? So there are different types of sensory organs according to what type of stimulus they receive. So first we have is mechanoreceptor. So the stimulus is usually mechanical. Let me give you an example. Like we have the receptor named LGC receptor, also known as nociceptor. These are the receptors for pains. They are the receptor for pain. They're usually present in the form of free nerve endings. So, you know, in your skin, a lot of free nerve endings are present and they receive the stimulus of pain. For example, someone prick you with a needle. At that time, the sensation of pain is picked up by these free nerve endings and they give information to the brain, right? Next, we have photoreceptor. These are for light. Like we have retina. Example, we have retina in our eye. So it contains certain photoreceptors like we have rhodopsin and we have iodopsin. Right? They contain this pigment. There are certain cells known as rods. These are photoreceptor cells and there are cones. These both they contain these pigment and they are receiving the stimulus, so you also call them as photoreceptors there. Okay, then we have chemoreceptors. They will detect the chemicals. In chemoreceptors we have an example of olfactoreceptors that are for smell, and then we have the gustatoreceptors that are for taste, right? Then we have thermoreceptors and these receptors are for temperature. These are for temperature. Fine. So various types of receptors are present in our body. So we have to study eye, ear and these olfacto and gustatoreceptors in detail. So we'll start with the olfactoreceptors first. So olfactoreceptors are responsible for smell. What are they responsible for? Smell. As you can see this epithelium is the olfactory epithelium. So this entire structure is olfactory epithelium. So this olfactory epithelium is present in the nose. Where it is present? In the nose. So here are certain cells present. These cells, these are modified bipolar neurons. These are your olfactoreceptor cells. So olfactoreceptor cells are the cells that will receive the stimulus. So what here is a stimulus? For example, someone cooked Maggi in your house. So the Maggi is being cooked in the kitchen, but you are sitting in your room. So you will get that sensation of smell just from there only. How? Because certain aromatic compounds, they get diffused from the Maggi, they get diffused into the air, and from air that chemicals or those aromatic compounds will enter into your nose. In the nose, there is a thick layer of mucus. This mucus is secreted by these Bowman's gland and the chemical will get mixed with this mucus and then it will bind to this structure which is the dendrite. Dendrite is a part of olfactoreceptor cell and this will causes depolarization or it will create action potential in this, or we can say these olfactoreceptor cell, they will get stimulated and through these nerve it will send the impulse to the olfactory bulb which will then send the impulse to the olfactory tract and from here it will go to the brain. Okay, and this is how you get the sensation of a Maggi. You have that memory in your brain that this is how a Maggi smell is. So a particular type of olfactoreceptor is present for a particular type of a smell that will stimulate a particular area in the brain. So this is how you can differentiate the smells or the olfactions also. Fine. Apart from that, supporting and basal cells are present. Basal cells are kind of stem cells. These are the kind of stem cells that will keep on multiplying, and form new olfactoreceptor cells. Okay, all right, so that's about your olfactoreceptor. And then we have this receptor known as gustatoreceptor cells. Gustatoreceptor cells, they are for taste. And where are they present? Definitely they are present in the tongue. In the tongue we have taste buds. So imagine this is one taste bud. Inside this taste bud is present your gustatoreceptor cell. Just like that of olfactory epithelium, it also has supporting cell and basal cell. Always the nerve endings are present in the sensory receptor cell, right? For example, the nerve ending was there in the was associated with the olfactoreceptor cell and here the gustatoreceptor cell. Same thing will happen here. There is also mucus in the saliva. This mucus contains food particle, right? And food particles have certain chemicals. For an example, if I say the sensation of salt or salty stuff. So in your food, to detect the salt, there is NaCl present, right? For example, in food, NaCl is present. So this NaCl will bind to this, your cells. Which cells? Gustatorreceptor cell, and it will stimulate these cells and they will send impulse to the brain. And this is how you can say, yes, the food contains salt. Yes, the salt is less. Yes, the salt is more. So depending upon the concentration of salt, your cells will be stimulated. If it is stimulated less, there is less salt. More cells are stimulated, more salt. Fine. Okay. Now let's talk about a question. The nose contains mucus-coated receptors which are specialized for receiving the sense of smell, are called olfactoreceptors. So answer is 2. Okay, next. The reflex pathway comprise of afferent neuron, both afferent and efferent, both afferent and efferent. Answer is 2. Always there should be, 3. Always there should be one sensory and one motor neuron. Okay, all right. Next. Next, we have the sympathetic neurons secrete dash near the visceral organs. So, we have done the autonomic nervous system. Parasympathetic always secrete ACH. Sympathetic secrete ACH in the ganglion, whereas adrenaline in the place of visceral organs. So, answer is 2. Okay. Next. Next, we have our another sense organ, that is eye. So, what's the function of eye and how many eyes do we have? We have one pair of eye. And where are they present? They're present in the socket or orbit of the skull. In the socket of skull. So the function of the eye is vision. Alright. So it's completely like circular in structure, as you can see. And it has 3 coats. How many coats does your eye have? I have 3 coats. So there are 3 coats in the eye. Okay. Now what are these? Let me just explain you. So there is an outermost covering, you call it as sclera. So from outside to inside we see the outermost covering of eye is sclera. Then inside you can see this blue colored coating, right? 3 layers can you see? Okay, if you cannot, let me just tell you. First this layer, then this blue layer, and then this one layer. There are 3 layers. If you can see clearly, there are 3 layers. So the outermost layer is known as sclera. The next layer is known as choroid, the middle layer, and the inner one is the retina, right? So sclera is also known as fibrous tunic. The choroid is also known as the vascular tunic, and retina is known as nervous tunic because it has a lot of neurons. This is known as fibrous because it has a number of fibers. What do it have? It have a number of fibers. Alright, so we'll see clearly this sclera, the outermost layer, is thicker at the posterior end, whereas when it is coming anteriorly, it has become thinner. And anteriorly it is thinner and it forms cornea. What does it form? Cornea. Okay. Similarly, the blue-black color layer is your choroid. Choroid is very thin posteriorly, just opposite to the sclera. It is very thin posteriorly, and anteriorly it forms first of all this ciliary body, and then it will form this iris. And iris is like a door. Iris is like the pigment, or we can say the color of the eye. Okay, iris is known as the color of the eye. For example, if I say I have brown eyes, so that means the color of my iris is brown. One very important thing: this ciliary body and your iris, both iris and your ciliary body, they both are muscular. They both are what? Muscular. They have muscles. Now, in between the iris is present this empty space, and this is known as pupil. This is the door to the eye. From there, the light enters into the eye. The light enters into the eye. Okay. Now, This ciliary body is attached to the lens. This structure is lens, right? But it is attached through these ligaments known as suspensory ligaments. They are present on both the sides. So once the question was asked in the NEET that how the lens are arranged or how the lens is attached with the ciliary body. It is attached with the help of suspensory ligament. So first there is lens. The lens, as you can see, it's biconvex, right? It's biconvex. It is attached to the ciliary body with the help of the suspensory ligaments. Now, here the lens is creating a wall and it has divided the entire eye into two chambers. This chamber is vitreous chamber. This vitreous chamber contains a jelly-like vitreous fluid. It contains vitreous fluid or vitreous humor, which is gel-like, right? The main purpose of this vitreous humor is to provide shape to the eye to keep it intact. Fine. Then here there is a space between cornea and lens, and that is aqueous chamber, and that contains aqueous fluid or aqueous humor. And aqueous humor is little water-like. It is little water-like. Okay. Now, moving further to the back side. Now there is a place, this place you can see, this portion particularly, let me just point it with the black one. This place is known as blind spot because at this place retina is not present, and retina is the innermost layer of the eye. Retina contains your photopigments which helps you to see. So there is a place, very small place where retina is absent. You call it as blind spot. What do you call it as? Blind spot. So at blind spot, no image is formed. At blind spot, no image is formed. No image is formed at the blind spot. Fine. And then through the blind spot, the optic nerve leaves and blood vessels enter. So that's the point where no image will be formed, but it is also a point from where the optic nerve will leave and blood vessels will enter. Okay, I will make you read NCERT of this very finely. Then there is a very small pit-like structure here. This is known as fovea, and it contains macula, which is a yellow spot. And I will let you know about this very soon. Okay. So first read the NCERT of everything that we have discussed about the basic structure. Retina will be discussing in the later of this lecture only. Okay. Okay, guys, let's see this one. Let's read eye. Eye is very important. That's why each and every line of the eye is important. So please open your NCERT books. Okay, that's the ear. All right, so let's get started. The adult eye is nearly spherical structure. As you can see, it's spherical. The wall of the eyeball is composed of how many layers? 3. The external layer is composed of dense connective tissue and is called sclera. I've told you it is known as fibrous tunic. Why? Because it has a lot of fibers. And which tissue have a lot of fibers? Connective tissue. The anterior portion of the layer is called cornea. So as I've told you, the outermost layer is thicker, but the anterior portion is thin and it forms cornea. Now, what is the function of cornea, guys? We'll take the NCERT side by side, okay? So what's the function of cornea? I said that sclera is, it consists of connective tissue, okay? And also the sclera forms cornea. The cornea is very thin, transparent, avascular, and a refractive body. That means it will allow the refraction of the eye. So whenever anyone undergoes LASIK surgery for removal of these specs, at that time the cornea's thickness has been changed and the refractive index is it's adjusted according to the number of the eye, right? Okay, because it is avascular, that's why it's very easy to do the operation and in fact the transplant also, right, guys? Also, this cornea is rich in collagen fibers. It is rich in collagen fibers. Okay, that's what I have told you. Let's see what NCERT has to say. Now, the middle layer choroid contain many blood vessels because it's a vascular coat. So definitely it will be having blood vessels and look bluish in color. Now, what's the color of the choroid? The color is blue. The choroid layer is thin over the posterior two-thirds of the eyeball, but it becomes thick in the anterior part to form ciliary body. So anteriorly it is thicker, just opposite to the sclera. The ciliary body itself continues forward to form a pigmented and opaque structure called iris, which is visible colored portion of the eye. So what gives color to the eye? Iris. Okay, then what's this white portion of the eye? That's sclera. So you heard me right? That sclera is the white of the eye. Then the eyeball contains a transparent crystalline lens which is held in place by ligaments attached to the ciliary body. In front of the lens, the aperture surrounded by the iris is known as pupil. The diameter of pupil is regulated by the muscles fiber of iris. So let's discuss the choroid in more detail and write this down. So choroid, what's the color of the choroid? Blue. Does it have a lot of blood vessels? Yes, it is vascular. So anteriorly it forms ciliary body, and ciliary body will later on forms iris, right? So if this is a ciliary body, this will be the iris. So this is the iris. Okay, this is the iris, and imagine this is also the iris. Okay, so if I say iris is also muscular, both these are muscular, so if they will, both these have muscles, if the muscles of iris will contract, so this space diameter can be changed. All right, so let's see what you see. Okay, so imagine this is the portion of eye that you see, like that. You see like that. Okay, imagine someone has blue eyes. If someone has blue eyes, so if the muscles of the iris contract or dilate, according to that, this surface area of the pupil can be changed. So this is how we say if the iris contract or it regulate, it can cause the pupil constriction or pupil dilation. If I'm talking about pupil dilation, that means the surface area is more. Imagine this is a situation where pupil is constricted. Okay, now let's see when there is a less light. When there is less light, the pupil is usually dilated. So pupil gets constricted when there is more light. When you for example, you are sitting in a room where there is no light. At that time, your pupils, they are dilated, but suddenly the light comes, you are not able to see properly. Why? Because a lot of light enters into your eye. Now your eye will adjust itself and it will constrict, right? So it will get constricted when there is more light. Okay, and when there is less light, the pupil will dilate. Right? That's about the choroid. Let's talk about your retina. Let's start with the retina, guys. Okay, so let's draw retina first. Let's draw the retina. First of all, imagine this is sclera. This layer is sclera. This layer is choroid. And now inner to this will be present the retina. So retina, first of all, it will be having a pigmented epithelium. You know why the choroid is blue in color? Because if it is darker in color, it will prevent the reflection of light and more light will be focused. Fine. So also retina have the same thing, just like that of choroid. That's pigmented epithelium. So this pigmented epithelium will prevent the reflection of the light. It will prevent the reflection of the light. Now comes the important photoreceptor cells. There are two types of cell. One that appears rod-like, so their name is rods, and other that appear like a cone, so their name is cones. Okay, so here we have cones and here we have rods. These both are what? These are photoreceptor cells. These are photoreceptor cells, and these cells will be having a photopigment. Now, because they are acting as a bipolar neuron, because as you can see their nerve endings like structures are present, they will be forming synapse with the bipolar neurons here. Okay, then comes the layer of bipolar neurons. There comes the layer of bipolar neurons. And then there will be a layer of ganglionic cells. They are also like neurons. They are a ganglionic cell. And then there will be optic nerve. Okay. Now from where the light will enter, for example, lens is present here. Lens, as you know, it's biconvex, made up of crystalline protein. From here, the light will enter. You heard it right. From here, the light will enter. If this was a structure of the retina, so the first thing that the light will come in contact will be your ganglionic cell. Then it will pass through these cells and then go to the photoreceptor cells for stimulation. Because ganglionic cells, they're not photoreceptor cells, and neither do the bipolar cells, they are also not the photoreceptor cell. So the light will pass through them and it will reach the photoreceptor cell to give the stimulus. Okay, now what will happen next? To know the next story, you should definitely know about rods and cones. Okay, so first of all, we'll talk about rods. Rods are the one that controls your scotopic vision. Scotopic vision or dim light vision, or moonlight vision, That means whenever you are in the low light condition, like in the moonlight outside during night, your rods will be functioning or working, and they are more in number. They are more in number than the cones. They contains a pigment. And the name of the pigment is rhodopsin. The name of the pigment is rhodopsin. For example, if this is rods, in this inner wall of the rods, these pigments will be present. So they are present in the inner walls. Okay. So these pigment, when the light struck on them, what happened to these pigment? Then how they are going to stimulate your brain? Let's see. So these pigments are generally made up of two things. One is retinal. Retinal is a derivative of vitamin A, which is retinol. Okay, what is retinol? Vitamin A. So vitamin A is formed in your liver from beta carotene, and then later on it gets converted into retinal, which is an important pigment of your rhodopsin and iodopsin both. That's why it is said that always eat carrot or vitamin A rich food. That's good for your eyes, right? And then we have the opsin protein attached to this. And in this case, the protein opsin is a scotopsin. Because opsin protein is different in both the pigments, this is how both the pigments are different, right? So like scotopsin is present with retinal, that's the rhodopsin. And the color of rhodopsin is purple. It is purple in color. Now what happened? How do you see? Now, for example, this retinal is always attached with this scotopsin. This is how they are married to each other. Okay. Now, when the light will come, this light will dissociate them. They will get separated. When the light will come, they both will get separated. Okay. And the purple color of this pigment will be converted into orange color. And you call this process as photo bleaching. Photon, light. Bleaching, that color has been removed. What does bleaching agents does? They remove the color, right? So just like that, here, photo bleaching has taken place. Now, if they have been dissociated, this will change membrane permeability. As I've told you, these pigments, they are present inside the membrane. This will change the membrane permeability and open up certain channels. So there will be change in membrane permeability. And there will be entry of sodium ions, because sodium ion channels open. Entry of sodium ions. And when the sodium ions enter inside the cell, what happens, guys? Depolarization. And hence the action potential has been done or created. And this is how this will send signal to bipolar neuron. And bipolar will send to ganglionic cell, And ganglionic cell, because all these are related to each other with synapses, this will send information to optic nerve, and via optic nerve this will go to occipital lobe of cerebrum. Okay. Now, this is the one, or this things happen whenever you use rods, and rods are usually responsible for dark vision, moonlight vision. So we can say that during darkness, depolarization takes place. Okay, or during dark condition. What about when you are using, when you are under light? So whenever you are under light, your cones are working. What are working? Cones. And cones never undergo depolarization, they undergo hyperpolarization. Okay, so whenever the hyperpolarization takes place, that means we are in the bright light condition. We are under the bright light condition. Okay, now let's talk about cones also. So this was about rods. So if I'm talking about cones, they are less in number and they are responsible for photopic or bright light vision. Photopic or bright light vision. Okay, so these cones, they have a pigment, and the name of the pigment is iodopsin. This iodopsin is made up of, first of all, retinal, and opsin protein. And this opsin protein is of three types depending upon the colors. Right, so you heard me right, colors. So cones is the one, it not only help you to see in the light, rather it also help you to see colors. So if ever or you are looking at this orange, pink, white, black color or blue color, this is just because of your cones. Okay, so now your opsins are of three types. One is erythropsin. Erythropsin is for the red pigment or red color. Whereas cyanopsin, what is the cyano word used for? Cyano word is always used for blue. And for green, we use the word chloro. So chloropsin is used for green. So if your green colored pigment is, stimulated, for example, it will be having a number of iodopsin, one with the opsin, green, chloropsin, one with the cyanopsin. So accordingly, what type of a pigment is stimulated, that will give you the sensation of that color. For example, I'm looking at the green color of the pen. That means here my green one or chloropsin has been stimulated. I'm looking at white. Okay, I'm looking at white. Here, all the three type of pigments are stimulated. So whenever you have a sensation of white color, all are stimulated. And whenever we are looking at black, nothing stimulated. Okay. Now, ma'am, what about the pink color, light blue color, and what about purple color? So as we have done in our junior classes, where that there are certain primary colors, you mix them and you can make another colors. So for example, two colors have been stimulated, that will give you a color of brown. So according to the stimulus and the power of stimulus, that different around thousands of different hues of colors can be seen. That's why girls know now. This is light pink, this is dark pink, this is baby pink, this is carnation pink, this is rose pink, right? So boys know only 2 or 3 colors. Girls know a lot. Okay. So this is what your eye is, and this is how it functions. Let's talk about the ear. So ear is the organ of what do you say? It helps in hearing. Okay. So what's the function of the ear, guys? Yes, it helps in hearing. And second, what's the another function? Okay, I think that color is not visible here. Maybe it's not completely stimulated. The pigments are not completely stimulated. So the function of ear is one, it helps to you to hear. And second, equilibrium and balance. Yes. So if we see the structure of ear, the ear is divided into three portions: the outer ear, the middle ear, and the inner ear, right? So from here to here is your outer ear. From here to here is your middle ear, and this pink thing entire is the portion of inner ear. Okay. So outer ear, middle ear, and inner ear. The outer ear consists of ear pinna, this opening which is external auditory meatus, external auditory meatus or the opening, and this long canal that is external auditory canal. And this external auditory canal have this have small hair and certain glands, small hair and certain glands, which are the wax glands. What are these? Wax glands or ceruminous gland. Wax or ceruminous gland. So these are the glands that you prick up with the help of your, what you say, earbuds or so. So next we have this thing, and this is known as tympanum. Tympanum or tympanic membrane is also known as eardrum. It is also known as eardrum. So in Hindi you call it as kaan ka parda. So it is always said whenever a little child is using earbud, parents usually prohibited them to use it. The reason is because they can, if they will put earbuds deeper in the ear, that can also damage this tympanic membrane. And then in the middle ear, you have this structure, which is the tympanic cavity. What is this? Tympanic cavity. This tympanic cavity have 3 ear ossicles, malleus, incus, and stapes. We have also discussed about them in the locomotion. And then there is a canal. What is this canal? Yes, have you ever heard of this tube? This is Eustachian tube. What is the function? What is the function of this Eustachian tube? See, from here the air is entering inside your ear and stops there because you have a drum here. An air cannot enter inside the middle ear. Fine. If you will just, if a nature would have not made any Eustachian tube and this middle ear would have been closed, there must be a pressure difference between this outer ear and middle ear. So we want to equalize the pressure on both the sides of eardrum, from the outer side and the inner side. So nature has given us the Eustachian tube. The Eustachian tube opens towards the pharynx. So whatever air you will engulf through the mouth, it will enter through Eustachian tube to the middle ear and provided the air, so that there is an equalized pressure on both the side of the tympanum. So what is the function of Eustachian tubes? It equalizes the pressure. It equalizes the pressure. Fine. And then there is a small window known as oval window. And this oval window opens to the inner ear. You call it as door to the inner ear. Door to the inner ear. So if we'll talk about the inner ear is present in the form of labyrinth. What is labyrinth? Bhul Bhulaiya, right? It is present in the form of labyrinth. So here, basically this structure is divided into 2 portions. This Mickey Mouse like structure is vestibule. Its function is in equilibrium. Whereas this round coiled portion is cochlea, and this one helps in hearing. Okay, so this is not what it is like. This inner ear is embedded in a bone, and the name of the bone is temporal bone. What bone? Temporal bone. Okay, let me show you a diagram. For example, if this is your vestibule, and this is your cochlea, this entire structure is present or embedded in the bony structure like this, right? The entire structure is present in the bone. So we also call inner ear as a labyrinth, or the other name of inner ear is considered as labyrinth. Okay, so this labyrinth is divided into 2 portions. One outer portion that is made up of a bone, and inside the membranous portion, which is made up of membranes. Fine. So if I take a section of it, for example, I'm taking a section of it like that, and I see it in this manner, I will see that outside present is a bony structure, this one. This is a bony labyrinth, and inside is present this pink color membranous structure, that is membranous labyrinth. And they have these spaces in between them, and these spaces are filled with fluid. You heard it right. These spaces are filled with fluid. The fluid present here is perilymph. Peri means outside. So that's a peripheral position. And this is the inside. So this is the endolymph. Okay. Let's talk about this cochlea in more detail. Okay, let's open the cochlea. Cochlea is just like a toy that you used to buy in your childhood when you used to go in fairs with your parents. There is a small toy that have a coiled ribbon like structure in front of it, and there is a whistle that is embedded to it, right, or attached to it. So when you blow air in the whistle, that coiled portion, it gets unwrapped or get uncoiled and it straighten up. So this is the same thing which we are doing. We are going to uncoil the cochlea. So if you uncoil the cochlea, what you will see, the outer portion which is made up of bone, right, and there is an oval window, and below oval window is present the round window like this. So if you'll see the structure here, you can see this is oval window and this window is round window. Okay, now, all right, so when I open this, the oval window opens on the upper surface. These are membranes. These are what? Membrane. These are two different membranes actually. The upper membrane is Reisner's membrane. What's the name of this membrane, guys? Upper membrane is Reisner's membrane. And the lower membrane is Basilar's membrane. The lower membrane is Basilar's membrane. Fine. And the outer portion is bony, and this is oval window. All right. So now this has divided the entire cochlea into three compartments. And now what are these compartments? The upper one's name is scala vestibuli, which have perilymph. This is scala tympani. This also have perilymph, and this one have endolymph, and its name is scala media, also known as cochlear's duct. Cochlear's duct. Okay. This basilar's membrane, it has certain organs present known as organ of Corti, which are the organs responsible for hearing. Now, how does organ of Corti looks like? Imagine this is basilar's membrane. Above basilar's membrane is present a glycoproteinaceous membrane known as tectorial membrane. What's the name of this membrane? Tectorial membrane. Okay. So let me just explain you one more time. On the basilar's membrane are present these small structures. You call it as organ of Corti, which have sensory receptors for hearing. Right? So this membrane is basilar's membrane. So if someone asks you, where does this organ of Corti present? Then what will you say? You will say they are present on basilar's membrane. Now on this membrane are present small cells known as hair cells. These hair cells, they are responsible for hearing. They contain this structure known as stereocilia. What are these structures? Stereocilia. And these stereocilia, they are also sometimes known as hair. Alright, and they are responsible for your hearing. How you will hear? Let's see. Now what happened whenever the sound enters? First of all, okay, let me just make it double so that your original diagram should not go waste. Fine. Okay. So whenever the sound enters, the ear pinna will collect the sound. Okay, something wrong. So ear pinna will collect the sound and it will enter from here, and it will enter into external auditory canal, and it will hit the eardrum. This eardrum, outside it is made up of a skin, and towards inside it is made up of mucous membrane. This will convert the sound waves into vibrations. It will convert these sound waves into vibration, and this vibration will then hit these small ear ossicle. These ear ossicle will now amplify that sound or vibration. The function of ear ossicle is to amplify the sound, right? So it will amplify the sound along with this oval window. Oval window will also amplify the sound. So remember, two things helps in amplification of sound. What are these? Amplification of sound. One, your ear ossicles. And second, yes guys, second is your oval window. Now your sound has entered into the oval window. Now it will go to the cochlea. All right, it will go to the cochlea. Imagine this is the oval window. Let me just multiply this also. Okay, so imagine this is the oval window. The sound will, all right, the sound will enter inside this. Okay, and it will move in this manner. Also, these vibration will go inside your cochlear's duct, and in the cochlear's duct, endolymph is present. So these vibration will move the endolymph, which will move the stereocilia. So once the stereocilia are moved, they will rub, they will get rubbed on the tectorial membrane, and when they will get rubbed on the tectorial membrane, they will get stimulated, and when they will get stimulated through this nerve, they will send signal to the brain. They will send signal to the brain. This is how they how you hear, right? So how you hear, guys? First of all, the sound will enter inside your ear, and once it will enter, it will hit the eardrum. Eardrum will convert into vibration. After that, ear ossicles and oval window will amplify the sound. So the main purpose of middle ear is to amplify the sound. Main purpose of the outer ear is to collect the sound and convert it into vibration. This is what tympanic membrane does. Tympanic membrane is a part of outer ear. Okay, then what is the function of inner ear? It contains the sensory cell. So if someone asks you, where does the hair cells present? Inner ear, outer ear, or middle ear? You will say inner ear. Okay. After it has entered through the oval window, now the vibration will go to the scala media. It will move the endolymph, and now endolymph will move the hair, and hair get rubs with the tectorial membrane, and the action potential is generated, which will go to the brain, and brain will interpret the sound. It will go to the temporal lobe of your cerebrum. Fine, guys? Now, let's talk about the balancing portion. Balance. How does we balance? So this is your vestibule. Okay, all right. Let me draw it again. So vestibule consists of this portion. These are semicircular canals. What are these? Semicircular canals. This is also semicircular canal. These semi means half circle. These canals are present at 90 degree to each other. They are present at 90 degree to each other. So at the base of every semicircular canal are present these swelling like structure. You call them as ampulla. What do you call them as? Ampulla. Okay. Now move further to the lower portion. In the lower portion is present two structure, utricle and saccule. Utricle and saccule. So utricle and saccule. Okay. All right. So ampulla and utricle and saccule, they contain the sensory hair cells that helps in balance. So ampulla contains the structure known as cristae. They contains cristae. These cristae contains hair cell which will help in the circular movement, right? Or rotational acceleration and deacceleration. Okay. Whereas utricle and saccule, these two contains utricle and saccule. They contain the structure known as maculae, and maculae helps in linear acceleration and deacceleration. They are usually responsible for your dynamic equilibrium, that means where you are moving, and they are responsible for your static equilibrium. Okay, they are responsible for your static equilibrium, linear acceleration and deacceleration. Static equilibrium, like you are sitting. Linear acceleration, deacceleration, that means you are in a lift. Okay, but you are static, but you are moving in the lift. And this one, it will responsible for your rotational acceleration and deacceleration. And they're responsible for your dynamic equilibrium. Okay, now how they help in balancing. So how does ampulla crystal looks like? They have these rows of cells known as hair cells, like this. Okay, and in between the hair cells are present certain supporting cells. What are present? Supporting cell. So these are supporting cells, and these are hair cell. So hair cells are important. They are acting as sensory cell. Above them are present the gelatinous layer known as scapula, or it's a gel-like fluid. Okay, a little between gel and fluid. So in the childhood, you must have heard when a gel-like fluid moves inside your head when you move, right? For example, I'm rotating. At this point, a gel-like fluid is moving. That's why when you stop, that everything is rotating, right? So this is gel-like fluid. So when you move in this manner, rotational acceleration, deceleration, at this point, this fluid move. This will lead to the movement of hair cells and hair, and it will, through nerves, send information to cerebellum. You heard it right? Cerebellum. Hearing information go to cerebrum, but equilibrium and balance is done by cerebellum. Now talking about macule. Maculae have small crystals of calcium carbonate known as otolith, and behind this is present otolith membrane, and this contains hair cell like this. Okay, now how does this work? These are certain supporting cells, and then there are certain hair cells. Okay, so this otoliths are made up of calcium carbonate. Otolith, they are made up of calcium carbonate. So for example, you are sitting in the car and suddenly someone push the brake, right? Or push the brake like that. So you will move your head like that. Okay, so at this point, when you move, your otoliths will move and they will get rubbed around the hair cell. Everything is related with rubbing of hairs of hair cells. Okay, and then action potential will be stimulated, or it will be produced, and the cells will be stimulated, and they will then send information to cerebellum, and then it will cause the muscular activity and bring you back to your position. This is how you can cause equilibrium and balance with the help of these organs. All right, guys. Okay, now it's time for certain, brainstorming thing, questions. Following are the functions of cones except daylight vision, scotopic vision, color vision, photopic vision. Okay, one more thing that I want to add in the eye. All right. So we have done this structure known as fovea. Fovea has a structure called yellow spot. So this is the area of highest resolution. This is the area of highest resolution. That means brightest image will be formed, and they only have cones. There are no rods. Okay, that's why it's the area of highest resolution, because it will help you to see all the colors and light clearly. So whenever you are making a brightest image, you are making the image on fovea. For example, just have a pen with you and look at the pen very clearly. Now you will see the outer, you're focusing on the pen, the brightest image formed in your eye or brain is your pens, and every other thing is blurred. Right? So that means you are producing the image on the fovea and every other thing is blurred. So this is like a portrait mode of iPhone, right? So, all right. So this was I wanted to add here. Anyways, let's talk about the questions. Back to questions. So following are the functions of cone except, yes, daylight vision. This is a function of rod, scotopic vision, and color vision and photopic vision. So answer is 2. All are the functions except 1. Next. In the human eye is composed of opsin, a protein, and retinal, which is the alcohol and derivative of vitamin A, aldehyde and derivative of vitamin A, aldehyde derivative of vitamin B, alcohol and derivative of vitamin B. So in your chemistry, you have studied anything which have all in it is an alcohol. Yes? Yes or no? Anything which have all, it is the alcohol, or anything which have ul in it is the aldehyde. So retinal is the aldehyde, and it's a derivative of, guys, it's a derivative of vitamin A, right? So aldehyde and derivative of vitamin A. Questions that are asked in the NEET, they're very basic. The most important thing in the zoology is that you always take it lightly. You think it's easy, and you started focusing on difficult things. But the questions that are always asked in the NEET, they're very basic questions, and you always get confused in the basic things only. So make sure you first sharpen up your basics and then go for the higher thing. Otherwise, higher thing is not even required in NEET zoology, I tell you. Okay. All right, next. The optic nerves leave the eye and the retinal blood vessels enter it at a point medial to and slightly above the posterior pole of the eyeball. It is known as. First of all, this is the eye. Okay. So it is talking about optic nerves leave, blood vessels enter. So from here you are already sure they are talking about blind spot. Now what about the location? Posterior pole it is a point medial to and slightly above the posterior pole of the eyeball. Okay, so it is saying that at a point medial to, medial means center, to and slightly above, this is the posterior pole, slightly above the posterior pole. So this is your blind spot. Okay, now lateral to the blind spot is present fovea. So if you'll read the NCERT, it has said lateral to it. Okay, lateral to it. I'll make you read that also. So here you can see the inner layer is the retina and contains 3 layers of neural cell from inside to outside, ganglion, bipolar, photoreceptor. There are 2 types of photoreceptor cell, rods and cones. These contains a light sensitive protein called photopigments. Okay, so let's move to that line which I am talking about. The optic nerve leave the eye and the retinal blood vessel enter it at a point medial to and slightly above the posterior pole of the eye. Photoreceptor cells are not present in that region and hence this is the blind spot. At posterior pole of the eye, lateral to the blind spot, there is a yellowish pigment called macula lutea with the central pit called fovea. So the full name of macula is macula lutea. Lutea is a word come from lutein. Lutein is a pigment, and wherever the word lutein comes, that means yellow in color. Okay, all right. So let's move there. And what's the name of the spot, guys? Blind spot. Answer is 3. And macula is also known as macula lutea. All right, next. Find the incorrect statement. The fluid-filled inner ear called labyrinth consists of 2 parts, the bony and the membranous labyrinth. Absolutely correct. The vestibular apparatus is composed of 3 semicircular canal and the otolith. This is also right. The vestibular apparatus contains these organs. Very true. Next. The malleus is attached to the tympanic membrane and the stapes is attached to the round window of the cochlea. This is true. Malleus is attached to the tympanic membrane, but stapes is attached to the oval window. That's why oval window helps in amplification. Eustachian tubes helps in equalizing the pressure on either side of the eardrum. This is also true. So answer is 3. Next. The next is a big thank you. So that's all about the neural. Most of the difficult parts of NCERT I have already covered. Other things I know you can easily read them out. So I have left it to you and I know you can do it well. So I'll meet in the next class with the chapter Chemical Control and Coordination. Till then, bye-bye. Take care. Lots of love. Bye-bye.